Welcome to Part 2 of the Doctors in Training 2019 Step 1 Review course. I'm Dr. Brian Jenkins, the founder and president of Doctors in Training. Part 2 of the course will review all of the high-yield topics you need to know to reach your USMLE Step 1 or COMLEX Level 1 exam goals. We know at this stage in your preparation, your main concern is crushing your exam, and we're going to help you do just that. Before we get started, I want to say a few words about how we've designed Part 2 of the course to optimize learning. First, long-term retention of information requires not only the input of information, but also the recall and application of that information. One of the reasons Part 2 of our course is so effective is that we ask questions to encourage you to recall and apply the information. Each Part 2 video begins with a warm-up review quiz to strengthen your retention of the medical knowledge we've already covered. And at the end of each video, there's an end-of-session quiz to review and reinforce the most important, most testable topics covered in that video. We've also teamed up with Lippincott, Williams & Wilkins, one of the most respected medical publishers, to provide you with more illustrations, diagrams, and clinical images than ever before. Let's take a minute or two to explain how Part 2 of the course is organized. You should have already received your Doctors in Training study guide. Your study guide is an integral component of Part 2 of the course. The study guide is where you'll find all the quiz questions and additional high-yield review material. If you open up the study guide to the table of contents, you'll notice that the course is divided into sections, starting with Foundations, Neuro, Pharm Basics, and so on. You'll also notice that within each section there's a chapter that corresponds to an individual video. So when you start watching the Foundations 1 video, you'll open the study guide to page 3, which is where you'll find Foundations video number 1. No matter what page you turn to in the study guide, you can always see exactly what video it goes with. The topic of the Foundations 1 video is Embryology. At the beginning of each chapter is a short outline of the material that is covered in that particular video. So on page 3, you can see that Foundations 1 will cover embryonic development, placental development, gastrulation, neurulation, and so on. And then there are some page references listed. Back on page Roman numeral 6, we have a key to abbreviations. We generally provide references to the last couple of editions of First Aid and a page reference to the most recent edition of Step Up to USMLE Step 1, as well as references to several other reference books, in case you need a more in-depth review of a particular topic. On page Roman numeral 8, you'll see the order we recommend that you view the course. We strongly recommend that you watch the videos in the order that they appear on your personalized dashboard. You have the freedom to view the videos in any order you like, but we've designed the course with a very specific order in mind for a couple of reasons. First, for starters, we're very deliberate about how we sequence the material in the videos, adding layers of information so that each video builds on the foundations of the previous videos. Second, the second reason for the recommended course order is that every video begins with a warm-up review quiz that reviews the high-yield material we've already covered in the previous portions of the course. Obviously, if you start going out of order, there's no guarantee that the warm-up material will be a review. You might find yourself getting frustrated rather than being reassured that you're actually learning and retaining everything. One of the things we have found to be true over the past decade is that a key indicator of success is to make a schedule that works for you and stick with it. So we strongly recommend that you develop a plan to work through Part 2 of the course if you haven't already done so. On page Roman numeral 7, there's a table titled Course Viewing Options. This table provides some different options for working through Part 2 of the course, depending on how many days you've set aside for Part 2 and how many hours per day you want to spend watching the videos. You can use the feedback button on the video viewer page to email us any suggestions or clarifications or mnemonics you want to share with us. We take your feedback very seriously, and your feedback is one of the ways we improve our course year after year. And while I'm thinking about it, one bit of feedback we receive from time to time is that certain lecturers are harder to hear than others, or that some students have a hard time hearing the heart sounds. So I want to point out that built-in computer speakers don't always reproduce low-frequency sounds very well. So if you're having any trouble with the video volume, we recommend that you try using some headphones. This year, we've also provided 2 diagnostic practice exams located at the bottom of the dashboard below the Part 2 videos. These practice exams will be available to you during your Part 2 access. We recommend that you take one diagnostic exam in the middle of the Part 2 videos and take the other toward the end of Part 2 to help you know how to focus your study time as you get closer to your exam date. We also recommend that you complete an NBME practice exam prior to starting the Part 2 videos and after completing Part 2 to provide clear benchmarks on your exam readiness. I also want to say a word about copyright and our terms of use. The entire Step 1 course, the diagnostic exams, the videos, and the study guide are all protected by federal copyright law. You may not copy any part of the course. In addition, our terms of use restrict someone from copying, distributing, selling, or sharing any part of our course or content. This includes sharing your Doctors in Training password with another person. In fact, sharing your course password can be grounds for criminal prosecution in accordance with the Computer Fraud and Abuse Act. Compliance with federal copyright law and our terms of use is very important. Copyright infringement is a criminal offense. It may also violate your school's ethics policy or honor code, and it could possibly make you ineligible for the residency match. Enforcement of this law and our terms of use helps to ensure that we are able to update our course each year so that it reflects the most up-to-date information. So if you see others stealing the course or sharing this course, please report the offense to our legal department at support@doctorsintraining.com. One last word of advice. As you work through the videos, it's often tempting to pause and rewind over and over again and to try to write down everything in the lectures. Don't. You'll end up using way too much time on the videos and you'll have trouble maintaining your pace. You're better off watching all of the videos and taking notes on the highest yield topics than to spend hours and hours taking meticulous notes on every little detail. You can always revisit the lectures later and rewatch a video that was really helpful. One last comment before we get going. Be sure to take good care of your body during this final period of intense study. You need to eat right and exercise regularly to relieve stress, and don't neglect adequate sleep, which is extremely important for memory processing so that you can remember everything you're learning. All right, let's get started.
Hi everyone, my name is Dr. Sherilyn Shakini. I'm a board-certified pediatrician, and I'm so glad that you've chosen to use Doctors in Training. Welcome to the Foundations 1 video. We're going to kick off the course with some basic embryology. Let's begin at the beginning, all the way back at conception. A sperm meets an oocyte, and that forms a zygote. The zygote begins to divide, and as more and more cells accumulate, it becomes a morula. Morula comes from the Latin word for mulberry, and the morula kind of resembles a mulberry. I like to know where some of these medical terms come from because I find that makes them a lot easier to remember. So we've gone from a zygote to a morula, and then as the morula keeps dividing and growing, it becomes a blastocyst. And the important thing about a blastocyst is that it has an inner cell mass and an outer trophoblast. These two parts of the blastocyst mark the first step towards specialization, where you take genetically identical cells and turn them into distinct, specialized tissues that perform specialized functions. The inner cell mass will eventually become the fetus, and the trophoblast will become the placenta. The placenta can't begin to develop until the embryo has implanted into the endometrium, which takes place on day 6. For that first week after conception, the zygote lives on nutrients that were stored in the oocyte. But eventually, the embryo needs more nutrients in order to grow and develop, and in order to obtain those nutrients from the endometrium, it needs a placenta. Implantation occurs when the blastocyst burrows into the endometrium. The trophoblast portion of the blastocyst further specializes into the inner cytotrophoblast and the outer syncytiotrophoblast. The syncytiotrophoblast is responsible for secreting the hormone HCG, which tells the mother's corpus luteum that a successful implantation has taken place, so that the corpus luteum will continue to secrete progesterone, and that progesterone maintains the endometrial lining and prevents menstruation. Later on, after implantation, the trophoblast eventually forms what we call the chorion, which grows little finger-like projections called villi, and these chorionic villi grow into the endometrium and become the fetal component of the placenta. The maternal component of the placenta is basically the endometrium that these chorionic villi are invading, and we call this maternal component the decidua basalis. And then, while we're talking about support structures, there's also a thin layer of tissue called the amnion, which contains the amniotic fluid. And there's something called the yolk sac, which serves as a secondary source of nutrients, kind of like the adult liver, as well as a source of non-specialized stem cells. While the trophoblast is undergoing specialization to become the placenta, the inner cell mass is also specializing and becoming a 3-layer disc called a gastrula. The first step toward becoming a gastrula is that during week 2, the inner cell mass becomes a 2-layer structure or a bilaminar disc. Those 2 layers are called the hypoblast and the epiblast. And that 2-layer bilaminar disc doesn't last very long, because during week 3, the epiblast will differentiate into a trilaminar disc with 3 distinct germ layers: the ectoderm, the mesoderm, and the endoderm. This trilaminar disc, again, is called a gastrula. Looking at this chart in your study guide, you can see how these 3 germ layers are basically going to become the entire fetus. We're going to go through these different cell types in just a second and talk about which tissues are derived from each of these 3 germ layers. But just to recap how far along we've come, we started with a single-celled zygote, which divided a few times and became a morula. Then the morula became a blastocyst, composed of that inner cell mass and the outer trophoblast. The trophoblast invaded the endometrium, and it will eventually become the placenta. And that inner cell mass becomes a bilaminar disc, and then a trilaminar disc with ectoderm, mesoderm, and endoderm. During weeks 3 through 8, which we call the embryonic period, these 3 germ layers begin to form organs. It's very important for your test to know which of these germ layers gives rise to these specific organs and tissues and individual cell types. You're very likely to be tested on the tissues that arise from the neural crest, which comes from the ectoderm. But first, we need to know what the neural crest is. The neural crest is involved with the very first steps of nervous system development, a process known as neurulation, or the formation of a neural tube. Here's how it works. By the end of the 3rd week of development, there are the 3 germ layers: the endoderm, the mesoderm, and the ectoderm. And right down the middle of the embryo, some of the mesoderm forms a little longitudinal cord of dense tissue called the notochord. The notochord's job is to induce the ectoderm cells right above it to form something called the neural plate. Then, once the neural plate has formed, the edges of it start to fold up and form this neural tube, which will go on to become the brain and the spinal cord. But during that folding process, some of these ectodermal neural plate cells kind of pinch off from the leading edges of the neural plate, and they merge to form a crest of tissue that overlies the neural tube. And this crest of tissue is called the neural crest. So the neural tube and the neural crest are derived from ectoderm, but it's the notochord in the mesoderm that makes it happen. Now, eventually, these ectoderm-derived neural crest cells are going to start to leave the neural crest and migrate to other parts of the embryo as it develops, like little ambassadors that perform various jobs throughout the body. Let's talk about what tissues and cell types are derived from the neural crest. The mnemonic we use is magic cops, like these neural crest cells are police officers that are dispersed in the body to do their various jobs. The M is for melanocytes. The A is for the aorticopulmonary septum, or the spiral septum, which separates the aorta from the pulmonary trunk in the developing heart. In cardiology, we'll review the embryology of the heart, and we'll talk about how if the aorticopulmonary septum fails to develop normally, you can get a truncus arteriosus, or transposition of the great vessels. The G is for ganglia, which includes autonomic ganglia, the dorsal root ganglia, which are the ganglia for the sensory afferent nerves of the body, and this also includes the enteric ganglia, the ganglia of the enteric nervous system. So the neural crest cells are basically responsible for the entire peripheral nervous system, and that also includes the Schwann cells. The I is for the stroma of the iris in the eye. We'll see that different parts of the eye are derived from all three germ layers. C is for chromaffin cells in the adrenal medulla. These are the cells that make catecholamines like epinephrine and norepinephrine. You can think of the adrenal medulla as being like a big, overgrown sympathetic ganglion. And we already noted that the sympathetic ganglia come from neural crest cells. The C in COPS is for the cranial nerves. Again, peripheral nervous system and ganglia, which includes the cranial nerves. The O is for the odontoblasts and also the ossicles of the middle ear. Odontoblasts are cells that play a role in the development of teeth, specifically the dentin of the teeth. So you might think about Crest toothpaste to remember that part of the teeth are derived from neural crest cells. The P is for the parafollicular cells of the thyroid, also called the C cells. These are the cells that secrete calcitonin. And then the S is for sclerae. You can also include Schwann cells here. So knowing all of these is a 3 or maybe a 4-star topic. Throughout the course, we'll occasionally give certain topics a star rating. It's just a shorthand way of telling you what you're most likely to be tested on. If we say something's a 3-star topic, that means there's about a 50% chance you'll see that on your exam. It's a topic that we know they test on, but only about 50% of students get a test item on it. A 4-star topic means that you should expect to see at least one question on that topic. Pretty much every student reports being asked about these 4-star topics. And then a 5-star topic means that you should expect multiple test items on that particular topic. So again, neural crest derivatives are a 3 or 4-star topic. There's better than 50% chance that you'll be asked to identify one of these cell types as a neural crest derivative. So I'm going to come back and review them in a slightly different way after we go through the rest of the germ layers. We talked about how the notochord induces the nearby ectoderm to become the neural tube, and how the neural crest splits off from the neural tube. So we have the neuroectoderm, which is basically the neural tube plus the neural crest, and that's distinct from the surface ectoderm. Let's finish the neuroectoderm first. We've already covered the neural crest, so now let's look at the neural tube. The neural tube basically gives rise to the central nervous system, which includes the neurons of the brain and spinal cord, the oligodendrocytes that myelinate those CNS neurons, the astrocytes that support those CNS neurons, and the ependymal cells that secrete CSF. The neural tube also gives rise to the pineal gland and the posterior pituitary or neurohypophysis, and also the retina. And then surface ectoderm is ectodermal tissue that did not become neurological because it didn't get influenced by the notochord. But still, I like to think of this as forming a bunch of other structures in the head, like the lenses of the eye, the olfactory epithelium, and the inner ear. So lots of stuff related to the special senses. Some other head structures that come from surface ectoderm are the anterior pituitary, the oral epithelium, the parotid glands, and the enamel of teeth. The rest came from the neural crest. And since this is the surface ectoderm, it's fairly easy to remember that the epidermis and the sweat glands come from surface ectoderm, and also the mammary glands. And one more, which is kind of an outlier, is that the portion of the anal canal distal to the pectinate line comes from the surface ectoderm. So both extreme ends of the GI tract come from surface ectoderm, the oral epithelium, and the distal part of the anal canal. Next, let's look at the structures derived from the endoderm. The endoderm is basically your gut tube, so most of these structures come from that gut tube. So there's the GI tract itself, from the esophagus all the way to the pectinate line in the anus. Where does the part of the anal canal distal to the pectinate line come from? From surface ectoderm. The submandibular and sublingual salivary glands come from endoderm. Remember, the parotid glands come from surface ectoderm, but the submandibular and sublingual glands come from endoderm. Other endoderm-derived tissues include the liver, the gallbladder, and the pancreas, also the lungs, the thymus, and the follicular cells of the thyroid. Also, make note, the lower portion of the vagina comes from the endoderm, and so does the epithelium of the bladder and the urethra. Most of the bladder and urethra are derived from mesoderm, along with the rest of the urinary system, but the bladder epithelium and the urethral epithelium are derived from the endoderm. And then the mesoderm is basically everything in between the ectoderm and the endoderm. It provides a lot of connective tissue and support tissues like muscle, bone marrow and blood cells, the heart, blood vessels, lymphatics, and the upper portion of the vagina. So the vagina is derived from both mesoderm and endoderm. The lower portion of the vagina is from the endoderm, and the upper portion is from the mesoderm. Also, the kidneys, the adrenal cortex, the gonads, and the dermis of the skin are derived from the mesoderm. So the bladder epithelium is endodermal, but the kidneys are mesodermal. The epidermis is ectodermal, but the dermis is mesodermal. That's a lot of information, and I know it can be overwhelming, but the highest yield part of this is knowing the structures that come from the neural crest. So take a look at number 2 in your study guide, and we'll review all of this in a slightly different way. It asks, which neural crest derivatives are found in each of the following adult structures? So I want you to pause the video and try to fill in as many of these as you can in about 1 minute, and then restart the video and we'll go over them together. In the peripheral nervous system, we get the cranial nerves, autonomic ganglia and nerves, the dorsal root ganglia and sensory nerves, and the enteric nervous system, and also the Schwann cells. In the ear, you have the ossicles of the middle ear, the malleus, incus, and stapes. In the eye, you have the stroma of the iris, and also the sclerae. In the mouth, you find the dentin of the teeth, which comes from the odontoblasts. In the thyroid, neural crest cells give rise to the parafollicular cells. In the heart, you have the aorticopulmonary septum. In the digestive system, you have the enteric nervous system. In the adrenal gland, the chromaffin cells of the medulla come from the neural crest. And in the skin, the neural crest gives rise to melanocytes. Make sure you know these. We're going to review more embryology later in the course. We'll talk about brain embryology and the pharyngeal apparatus in the neuro section, and we'll talk about cardiac embryology in the cardiovascular section and so on. But I do want to highlight a few important dates related to organogenesis, just to give you some big picture markers. We call weeks 3 to 8 the embryonic period, and this is the time when organogenesis takes place. This is the crucial time period in which the embryo is most susceptible to teratogens during organogenesis. Week 4 is when the 4-chamber heart begins to develop, and also when the 4 limb buds begin to form. So that's easy to remember. Week 4, 4-chamber heart, 4 limb buds. Week 8 is when we start to see fetal movement, and you can use the fact that there are 8 letters in the word movement to associate fetal movement with week 8. And one student suggested that you remember that 8 rhymes with gait, and that will help you think about fetal movement. It's not like the fetus is walking around inside the uterus or anything, but that might be a helpful mnemonic. And week 10 is when the genitalia begin to take on visible sex-specific characteristics. You can think about puberty starting around age 10, or you can think of the word tenitalia, whatever works for you. And remember that these weeks are being measured from the day of conception. In embryology, we're talking about developmental age, which is measured from conception. That seems fairly obvious, except that OB-GYNs in the US keep track of gestational age. They count weeks of gestation from the last menstrual period, not from the day of conception. So a 40-week gestational pregnancy in the US is actually a 38-week-old fetus by developmental age. Now we're ready for the end of session quiz. At the end of each video, we're going to have a short quiz about 3 questions to review some of the highest yield material covered in that video. The quiz is found in your study guide, so what I want you to do is pause the video for a minute or 2 and try to answer questions 3, 4, and 5 in your study guide. Don't spend too long on them. Then once you've answered those questions, you can restart the video and I'll go over the answers. All right, let's go over the answers to the end of session quiz together. First question, what is the relationship between the notochord, the neural crest, the neural plate, and the neural tube? The notochord is part of the mesoderm, and the notochord induces the nearby ectoderm to form the neural plate. Then the neural plate gives rise to the neural tube and the neural crest cells. Next, what is the embryologic origin of each of the following adult structures? Anterior pituitary arises from surface ectoderm. Posterior pituitary comes from the neural tube. The parotid glands come from surface ectoderm, but the sublingual salivary glands come from endoderm. The sclera comes from the neural crest. The lens comes from surface ectoderm. The retina comes from the neural tube. And the mammary glands and sweat glands come from surface ectoderm. We're going to quiz you on these several times throughout the videos. And the last question, at what developmental age does each of the following events typically take place? 4-chamber heart begins to develop. That's 4 weeks. Remember, 4-chambered heart, week 4. Genitalia begin to take on visible sex-specific characteristics. That's at 10 weeks. Fetal movement begins. Movement has 8 letters, so that's 8 weeks. Limb buds begin to form. 4 weeks, 4 limbs, week 4. That's it for the end of session quiz. The next video is Foundations 2. We're going to talk about teratogenesis. See you then. Welcome to our video on teratogenesis, or as I like to call it, embryology gone wrong. We're going to discuss teratogenic drugs first and some environmental exposures that can be teratogenic, and then finally we'll talk about some genetic things that can cause congenital malformations. In the last video, I mentioned that there is a period of development during which the baby is most susceptible to teratogens. Do you remember when that is? It's the embryonic period, which is weeks 3 to 8 of development. And why is this such a bad time for an embryo to be exposed to teratogens? What's going on during this time frame that's so crucial? Organogenesis. If you damage a developing organ or prevent it from developing normally, that's more likely to cause congenital defects. So let's start with some teratogenic drugs. Take a look at your study guide. This isn't meant to be a complete list by any stretch of the imagination, but it's a good start. The first one is ACE inhibitors and ARBs, which can cause renal failure and oligohydramnios. We use these drugs to protect the kidneys in diabetic patients, so it's easy to remember that they can cause renal damage in the fetus. There are 3 big classes of antibiotics that you should know about. Aminoglycosides, like gentamicin, can cause ototoxicity and cranial nerve 8 damage. Fluoroquinolones, like ciprofloxacin or levofloxacin, have a theoretical risk of causing cartilage damage in the fetus and also in kids based on animal studies. So we generally try to avoid quinolones in pregnancy, although there isn't good definitive data that they actually cause any problems in humans. And then the third group of antibiotics is the tetracyclines, like tetracycline and doxycycline, which can cause discolored teeth in kids. So in general, we don't give tetracyclines to children for that reason. And this could happen in utero as well. Another group of drugs that are potentially dangerous are cancer drugs. Cancer drugs obviously interfere with rapidly dividing cells, so that's a bad thing if you're an embryo that's trying to make organs. Cyclophosphamide and other alkylating agents can be particularly teratogenic. They can cause ear and facial anomalies, limb hypoplasia, and absence of digits. They're obviously going to interfere with rapidly growing cells. And then another cancer drug that's noteworthy for being teratogenic is methotrexate, which is a drug that works by interfering with folate metabolism, and that can lead to neural tube defects. We'll see that again in neuro when we talk more about the development of the CNS and neural tube defects like spina bifida. But the reason we give pregnant women prenatal vitamins that contain a large dose of folic acid is to prevent neural tube defects. So lock those two things together in your mind: neural tube defects and folic acid. And one of the approved uses of methotrexate is to terminate an ectopic pregnancy. So obviously you don't want to give that to a pregnant woman who's trying to stay pregnant. And you're probably inclined to say, gosh, if you're getting chemotherapy for cancer while you're pregnant, that's a pretty bad deal no matter what. Which is true, but I just want to point out that both of these drugs, cyclophosphamide and methotrexate, aren't just used for cancer. They're also used to treat inflammatory diseases like lupus and psoriasis and rheumatoid arthritis. So even though I've grouped these together as cancer drugs, that's not the only time you might see them used. The next few drugs in our list are seizure drugs. Both carbamazepine and valproic acid are known for causing neural tube defects, just like methotrexate. So if you have a patient with a history of seizures that's on one of these drugs, you want to make sure she's getting that folic acid supplement for at least a couple of months before she starts trying to get pregnant. And if possible, you want to get her off of the seizure drug, but sometimes you just can't. We used to recommend giving extra high doses of folic acid to these women, but that's never actually been proven to be beneficial. And then another seizure drug to mention is phenytoin, which can cause something called fetal hydantoin syndrome. That'll be covered in the neuro section when we talk even more about seizures and seizure drugs. Lithium is used as a mood stabilizer in patients with bipolar disorder, but it can cause a cardiac malformation called an Epstein anomaly. We'll talk about that in the cardiology section. Atorvastatin and other statin cholesterol drugs are basically contraindicated in pregnancy because they interfere with cholesterol synthesis, which is really important for making a lot of steroid hormones and other things the developing fetus needs. So there's a possible increased risk of CNS and lib abnormalities. Another medication you should know about is warfarin, which primarily can cause bone and cartilage problems, especially in the epiphyses. It can also cause nasal hypoplasia, which is kind of unusual. Which anticoagulant is considered safe during pregnancy? Heparin. Be sure you know that distinction. Isotretinoin is a retinoid, a vitamin A derivative. Vitamin A is retinoic acid, right? Isotretinoin is contraindicated in pregnancy because it causes lots of spontaneous abortions and a fair number of birth defects, like skull and CNS problems, heart defects, or parathyroid gland problems. There's a classic teratogen called diethylstilbestrol, or DES, that's so bad it was taken off the market. Children that are exposed to DES in utero are at high risk of developing vaginal clear cell adenocarcinoma later in life, often in their teens and 20s, but sometimes as early as 7 or 8. The other classic teratogen that nobody uses much anymore is thalidomide. Thalidomide was a sedative that was given to pregnant women 50 to 60 years ago until it was found to cause limb deformities called phocomelia, where you have severe hypoplasia of the arms and or legs. Thalidomide can be used to treat Hansen disease, also known as leprosy, but when was the last time you saw a patient with leprosy? And it's sometimes used to treat multiple myeloma, but that's usually a disease of older people well past childbearing age. Next, let's go over some other types of teratogens besides drugs. Some of these are kind of hard to avoid, but they're worth knowing for your test. Ionizing radiation, like X-rays and CT scans, can cause CNS malformations if the exposure occurs early in pregnancy, and an increased risk of cancer, especially leukemias, later in pregnancy. We generally try to avoid X-rays during pregnancy, but sometimes it's impossible. Excessive amounts of vitamin A can be teratogenic, just like we said with the vitamin A derivative, isotretinoin. It causes spontaneous abortions, microcephaly, and cardiac defects. Excess vitamin A can also interfere with the migration of neural crest cells, and it can interfere with homeobox gene or HOX gene expression. We'll talk about HOX genes and what they do in just a moment. Maternal diabetes can cause all sorts of problems like cardiac defects and neural tube defects. It can also cause something called caudal regression syndrome, where the sacrum fails to develop normally. Not gestational diabetes, because that happens way after the embryonic period and organogenesis. We're talking about women with pre-existing diabetes who then become pregnant. Along the same lines, maternal iodine deficiency can cause the baby to be born with congenital hypothyroidism, which we call cretinism. In this case, it's not the iodine that's teratogenic, it's the iodine deficiency that causes problems. Likewise, nicotine and cocaine aren't necessarily teratogens per se. They don't interfere with organogenesis, but they can lead to placental abruption, low birth weight, and intrauterine growth restriction because they interfere with the blood flow to the fetus. And there can also potentially be some neonatal addiction to these substances and withdrawal symptoms in the first few hours to days of life. But alcohol, on the other hand, is a major cause of birth defects. So let's talk about fetal alcohol syndrome for a moment. Excessive alcohol consumption, either binge drinking or lots of drinks on a daily basis, can be harmful. But we don't really know what a safe amount of alcohol would be to consume during pregnancy, so the Surgeon General recommends no alcohol at all during pregnancy. What do you see with fetal alcohol syndrome? There are some characteristic facial abnormalities with fetal alcohol syndrome, which include narrow eye openings with wide-set eyes, a smooth philtrum, and a thin upper lip. Alcohol can also cause cleft lip and cleft palate, so lots of midline facial abnormalities. Fetal alcohol syndrome can also cause intellectual disability, which used to be called mental retardation. And along with that, you can have microcephaly. You can also see something called holoprosencephaly. Holoprosencephaly is where the hemispheres of the brain fail to separate across the midline. And often it's associated with facial anomalies where the facial structures fail to separate, and you can get a single midline eye. So again, another kind of midline facial deformity. So those are all the teratogenic drugs and exposures we're going to cover. I know it's a lot of material, so let's go to the whiteboard and review these in a slightly different format. All right, I want you to take out a piece of paper and write down these 7 different anomalies that I've listed over here on the left, and then we're going to match them to the various teratogenic drugs I've listed on the right. Don't get lazy and just watch me do it. It's really helpful to go through the effort to write these down because it forces you to process this stuff using another part of your brain. Cartilage damage pairs with levofloxacin. Remember, fluoroquinolones hurt the bones. Discolored teeth pairs with tetracyclines. Epstein anomaly, the cardiac anomaly, where the tricuspid valve is displaced downward into the right ventricle so that the right ventricle is hypoplastic and the right atrium is abnormally large, is associated with use of lithium. Holoprosencephaly, where the brain hemispheres don't separate, can be associated with maternal ethanol use. Next is limb hypoplasia. Lots of things can do this, but we mentioned it with thalidomide and also cyclophosphamide. Spina bifida. Remember, spina bifida is a neural tube defect, and we said when you hear neural tube defect, think folate deficiency. But the drugs listed here that also cause neural tube defects are the 2 anti-epileptics, carbamazepine and valproic acid. And the last anomaly here is autotoxicity, which can be due to aminoglycosides, and gentamicin is the only aminoglycoside listed here. Be sure you can recognize these drugs as teratogens and know the problems that they are most likely to cause. And as a little treat for you today, I have a gold star. We're going to wrap up this video by talking about several genes that are crucial for normal embryogenesis and how mutations in these genes can lead to congenital anomalies. I just want to highlight a couple of the more testable ones. The first genes I want to mention are the homeobox genes or HOX genes. There's a short section on the HOX genes in your study guide. Homeobox genes are the blueprints for skeletal morphology. they code for transcription regulators, so these gene products regulate the expression and transcription of other genes. Basically, Hox genes make sure that the skeleton is laid out correctly, so you don't have arms and legs in the wrong places. There are some experimental models in which they've altered the Hox genes in fruit flies, and they end up growing legs on their heads where the antennae should be, or an extra set of wings, that sort of thing. In humans, mutation of the specific homeobox gene HOXD13 causes something called synpolydactyly. Poly is many or multiple, and dactyly has to do with the digits. So polydactyly is when you have more than the normal number of digits on a hand, like the six-fingered man in The Princess Bride. But then syn means together, so syndactyly is where you have fusion of the fingers. So with this HOXD13 mutation, you have synpolydactyly. So there are extra digits and they are fused together. As we've already mentioned, excess vitamin A, or retinoic acid as a derivative of vitamin A, can alter Hox gene expression. So again, we don't give vitamin A supplements to pregnant women. The next gene to discuss is the sonic hedgehog gene. This family of genes was first discovered in fruit flies, and for whatever reason, they were named hedgehog genes. Then somebody got cute and named one of the genes Sonic Hedgehog, like the video game character. There's also a cancer-causing gene that they named Pokemon until Nintendo threatened to sue. True story. But this sonic hedgehog gene is expressed at the buds of the limbs in a zone called the ZPA, or the zone of polarizing activity. And kind of like the Hox genes, the sonic hedgehog gene is involved in organizing the layout of the embryo in an anterior to posterior direction, setting up which pole is going to become the head and which pole is going to be the tail, and then organizing all the points in between. One thing you need to know is that mutations of sonic hedgehog can cause holoprosencephaly, where the hemispheres of the brain fail to separate across the midline, and in severe cases, you have that single midline eye. Where did we see that earlier? That's right, fetal alcohol syndrome will also cause this. So sonic hedgehog is important for organizing the embryo along the anterior-posterior axis in the craniocaudal direction. And there's another gene called WNT7, which is important for organizing the embryo along the dorsal ventral axis so that your feet and your nose are pointing in the same direction. One student suggested that the NT in WNT7 could remind you that this gene controls the direction of the nose and toes. And there's a gene called FGF that is important for limb lengthening. FGF stands for fibroblast growth factor. So if you have a defect of the FGF gene, you're going to have short limbs. And actually, a mutation in the gene for the FGF receptor causes achondroplasia, which results in, you guessed it, short limbs. All right, that's it for our video on teratogens. Go ahead and pause the video and answer the end of session quiz questions on your own, and then we'll go through the answers together. Let's go over the answers to the end of session quiz. First question, which antibiotics are potentially teratogenic? Aminoglycosides, tetracyclines, which cause discolored teeth, and fluoroquinolones, which can potentially cause cartilage abnormalities. Next question. A pregnant woman is diagnosed with a deep venous thrombosis of the left leg. Which anticoagulant is contraindicated during pregnancy? That's warfarin. And which anticoagulant is preferred? That's heparin. And the last question. Examination of a fetus from a spontaneous abortion at 14 weeks gestation reveals a vestigial pair of legs that arise from the torso just below the arms. A mutation in what family of genes is the most likely cause of this malformation? This is the homeobox genes, Hox genes. Remember, Hox genes determine the layout of the appendages. That's the end of session quiz. But just below, you'll see this table called Rapid Fire Facts. We have identified almost 300 of the highest yield factoids and high yield associations, most common causes, buzzwords basically. A lot of people will tell you that step 1 doesn't test buzzwords, but honestly, if you know the buzzwords, it will save you some time in coming to the right diagnosis quickly, so that then you can answer whatever the question is asking about the pathophysiology or whatever. So in some of our videos, after the end of session quiz, we'll have a handful of these rapid fire facts for you to jot down. Then later you can flip through these and cover up one side and quiz yourself, or get a study partner and take turns quizzing each other. These are great to go through at the end of the course, maybe on the last day before your exam. So let's go through these rapid fire facts. The most common cause of neural tube defects is folate deficiency. The most common preventable cause of congenital malformation in the U.S. is alcohol use during pregnancy. All right, that's it for this video. I'll see you next time.
In these next few Foundations videos, we're going to switch gears and review some basic cell biology. Now, I'm sure you're familiar with a lot of this already, but you may not have thought about it much lately, so we're going to move through a lot of this pretty quickly just to jog your memory and to focus on the cell bio topics that have a tendency to show up on step 1. The first topic we're going to review are the various cell organelles, starting with the nucleus. Now, the nucleus is contained by a nuclear envelope, and it's a very tightly regulated structure. The nuclear envelope is kind of like a bouncer that guards the nucleus and keeps everybody out, because not everyone gets to party with the DNA. In fact, so the nuclear envelope is like a bouncer at a club guarding the door. He keeps the DNA inside the nucleus, but he also has to let the mRNA that's transcribed out of the nucleus. So what determines what can enter and leave the nuclear envelope? Well, the nuclear envelope has nuclear pores, and the only way to get through the nuclear pores is by having something called a nuclear localization signal. These are definitely worth knowing for your test, so let's take a look in the study guide. The nuclear localization signal is a specific sequence of amino acids consisting of 4 to 8 amino acids. It's rich in the amino acids proline, arginine, and lysine because these are more positively charged amino acids, which allows them to bind more readily to the negatively charged DNA. These nuclear localization signals are present on proteins that are headed for the nucleus, like histones, because the proteins are synthesized in the cytoplasm and then transported to the nucleus. The nuclear pores recognize these signals and allow the proteins to enter the nucleus. So if the nuclear envelope is like a bouncer that regulates who gets into the nucleus, the nuclear localization signals are like a password to get those nucleus-bound proteins past the bouncer. So last year, a student suggested that to get into the club, you have to be pals with the bouncer. And PAL stands for proline, arginine, and lysine, which are the amino acids found in the nuclear localization signal password. And another student suggested that the bouncer only lets people who bring positive energy into the club, because again, those are positively charged amino acids. Then one last point. If you have a mutation in a single amino acid in this 4 to 8 amino acid sequence, it could potentially prevent that protein from being transported into the nucleus. While we're talking about the nucleus and DNA, let's talk about the different phases of the cell cycle. Right after a cell has completed mitosis, it goes into interphase. And the first part of interphase is called the G1 phase, for growth phase 1, or gap phase 1. So cells that are designed to divide very rapidly, like bone marrow stem cells, have a fairly short G1 phase because they want to get to S phase quickly, because S phase is where the DNA is replicated. So S is for synthesis of DNA. Then once a cell gets to S phase, it's committed to cell division. But some cells are not meant to divide, so they actually go into a G0 phase. And then cells like hepatocytes and lymphocytes are on standby, and they can go from G0 to G1 in order to grow and prepare for mitosis if needed. Then after the G1 growth phase, there's S phase, and then there's a second growth phase called G2, and then an M phase where mitosis takes place. But our bodies need to be able to control and regulate the cell cycle to keep cells from just growing and dividing out of control. So there are checkpoints along the way that control the transitions between these different phases. Now, these transitions are regulated by 3 things mainly: tumor suppressor proteins, cyclins, and cyclin-dependent kinases. So let's start with a few tumor suppressors. Going from G1 to S phase, there's the RB gene and the P53 tumor suppressor gene. So both of these genes encode protein products that are critical for regulating the transition from G1 to S phase. If these proteins don't give their stamp of approval, then the cell isn't allowed to go into S phase. Now, mutations in these genes result in unregulated growth and cell replication, which is why these are called tumor suppressors. As long as these genes and their protein products are functional, they suppress the unregulated growth that we see in cancer. Now, there are two main cancers associated with mutations in the RB gene. We'll talk about this in detail in the oncology section, but one cancer associated with this is retinoblastoma, which is why this is called RB. The other cancer is osteosarcoma. And then P53 is also involved in regulating the transition from G2 to M phase. Next, let's talk about cyclins and cyclin-dependent kinases, or CDKs. A kinase is an enzyme that uses ATP to phosphorylate things, so to add phosphate to things. Kinases are expressed constantly. They're always around, but they are inactive unless they need to be active. Now, cyclins are regulatory proteins that control cell cycle events. They activate those cyclin-dependent kinases. Now, in order for the cell cycle to progress, these CDK and cyclin complexes have to be activated and then inactivated. Let's look at the study guide and dig into the details about how these work. Cyclins and CDKs phosphorylate target proteins to drive the cell cycle. All cyclins are degraded by ubiquitin protein ligase when their cell cycle specific job is complete. Ubiquitin is a protein that gets tagged onto other proteins to mark them for destruction. If a protein gets tagged with ubiquitin, that's a signal for this ubiquitin protein ligase to break down that protein. So because of this, ubiquitin is a very important molecule in regulating the cell cycle. You need ubiquitin in order to shut down these cyclins. Next, tumor suppressors P21, P27, and P57 bind to these cyclin CDK complexes and inactivate them. We mentioned P53 before, and P53 controls the transcription of P21. So tumor suppressors inactivate cyclin complexes and prevent them from continuing to phosphorylate things. Again, because these kinases are always around, we need to be able to activate and inactivate them at precisely the right times. So going from the G1 to the S phase, there are a few specific molecules that are worth knowing. You have cyclin D that binds and activates CDK4. Then CDK4 phosphorylates the RB protein, which allows that RB protein to be released from a transcription factor called E2F. Now with E2F unbound, the cell's free to transcribe and synthesize components needed for progression through the S phase. So those components are things like DNA polymerase, thymidine kinase, dihydrofolate reductase, and cyclin E, all of which are needed in order to synthesize DNA. And you can't get these components unless E2F comes unbound from RB, which only happens when cyclin D and CDK4 are around. Now this is a pathway you need to know for your test. It's a picky little detail, but you need to know it. Then the next bullet says that cyclin E binds and activates CDK2. And when this happens, the cell is allowed to progress into S phase. So we see two regulatory components that allow the cell to progress from G1 into S phase. CDK4 coupling with cyclin D, and cyclin E coupling with CDK2. So make sure you pair those up in your mind. One student suggested that you can remember CDK4 goes with cyclin D because D is the fourth letter of the alphabet. But however you do it, make sure you remember those. Then going from the G2 phase into M phase, cyclin A and CDK2 allow the cell to progress into prophase of mitosis. And then the cyclin B and CDK1 complex is activated by CDC25, which causes the breakdown of the nuclear lamins and the initiation of mitosis. Nuclear lamins are basically the skeletal framework of the nucleus. It's a type of intermediate filament, and the skeletal framework has to be broken down in order for the nucleus to divide and for the cell to divide. That's one process that regulates mitosis. The cell nucleus holds the DNA that codes for the proteins that cells produce, but it's the proteins that actually do all the work. So let's talk about the cellular machinery that cells use to synthesize and process these proteins. We're going to be looking at some electron micrographs because you need to be able to recognize these various organelles. You're very likely to have a question asking you to identify a particular organelle. It's probably a 4-star topic, which means nearly everyone can expect at least one question on this. So let's start with the rough endoplasmic reticulum, or the rough ER. The rough endoplasmic reticulum is rough because it has ribosomes. Now the ribosomes are translating a lot of protein. Specifically, this is where we synthesize the proteins that will be secreted outside the cell, and the place where end-linked oligosaccharides are added to the proteins. So it makes a lot of sense that cells that export a lot of proteins, like plasma cells, have a lot of rough endoplasmic reticulum. So where in the cell do we make the proteins that are going to remain in the cell? Well, there are also free ribosomes suspended in the cytoplasm that are not attached to the rough ER. And these free ribosomes are the site of synthesis of cytosolic proteins and the proteins that become part of the organelles. Now, in the neurons, the rough endoplasmic reticulum is called Nissl bodies or Nissl substance. And the Nissl bodies synthesize enzymes and peptide neurotransmitters. Now, these Nissl bodies are found in the dendrites, but not in axons. The axon hillock sometimes looks very similar to a dendrite up close. So if you look at the cell body and you see Nissl bodies, you know you're looking at a dendrite rather than an axon. And then in addition to rough endoplasmic reticulum, there's also smooth endoplasmic reticulum. It's like the Justin Timberlake of cell biology. Smooth. Smooth endoplasmic reticulum is where steroids are synthesized and where drugs and poisons are detoxified. So where would you expect to see a lot of smooth endoplasmic reticulum? In the adrenal cortex and the hepatocytes. So the adrenal cortex cells are rich in smooth ER because they're making steroid hormones. And hepatocytes are important for metabolizing and detoxifying things. So if you take a lot of medications, your hepatocytes are going to have a lot of smooth endoplasmic reticulum in order to metabolize all those drugs. So going back to the synthesis of proteins, once we've translated the amino acid sequence in the rough ER, we need to make a bunch of post-translational modifications to that peptide. And those modifications take place in the Golgi apparatus by and large. But how do we get the new polypeptides over to the Golgi? You don't just turn them loose and cross your fingers and hope they find their way to the right place. Instead, our cells use something called trafficking proteins, which help the endoplasmic reticulum form vesicles that can carry the proteins to the Golgi. So sometimes you'll see these called vesicular trafficking proteins. So there are three basic trafficking proteins that I want you to know. The first two are called coat proteins because they coat the surface of the endoplasmic reticulum or the surface of the Golgi. And then when it's time to form a vesicle, the coat proteins form spherical polymers that sort of help shape vesicles into little spheres so that you can move the proteins around inside the cell within those vesicles. So there's coat protein 2, or COP2, which is involved in the anterograde trafficking, moving protein products and steroid products forward from the ER to the Golgi apparatus. And then the COP1 proteins are involved with retrograde trafficking of vesicles, where stuff is moving from the Golgi back to the ER. So usually proteins go from the ER to the Golgi and then on outside the cell. But sometimes you need to ship something backwards from the Golgi back to the ER. And if you want to go backwards, you need a COP1 protein. And then the third trafficking protein is something called clathrin, which coats the far side of the Golgi, the trans side of the Golgi. And it helps it form vesicles to transport enzymes called hydrolases to the lysosomes. The lysosomes are little vacuoles full of enzymes that break down cellular debris and waste products. So lysosomal hydrolase actually do all the breaking down, but they're synthesized inside the rough endoplasmic reticulum, and then they're transported to the Golgi for modification, and then once they're all ready to go, the clathrin helps the Golgi form these little vesicles to move the hydrolases out to the lysosomes. Then the other big job of clathrin is to help the plasma membrane bring stuff from outside the cell into the cell. But we're going to talk about that in Foundations 5 when we talk about the plasma membrane. So we've made our proteins in the rough endoplasmic reticulum. We used COP2 trafficking proteins to move it over to the Golgi apparatus. The Golgi apparatus does a lot of modification and packaging of proteins and lipids and other secretory products to get them ready for export. Now the functions of the Golgi apparatus can be divided into two big categories. The first category is the distribution of proteins and lipids from the endoplasmic reticulum to the plasma membrane, and also the lysosomes and the secretory vesicles. So the Golgi apparatus is kind of like the cellular post office. It makes sure that everything gets sent to the right place. In order for a protein to get to the lysosome or to get packaged in a secretory vesicle, it has to go through the Golgi. The second big job of the Golgi apparatus is post-translational modification of proteins and proteoglycans, which are just proteins with saccharides or sugars tacked on. Proteoglycans are an important part of connective tissue. Now there are lots of different types of post-translational modifications going on here. The Golgi glycosylates core proteins to form proteoglycans, tacking things like chondroitin sulfate and heparin sulfate onto the core proteins. The Golgi also adds sulfonyl groups to sugars in proteoglycans and to selected tyrosine residues in proteins, and this is called sulfation. It can add O-oligosaccharides to serine and threonine residues. And then the last modification that I really want you to know is that the Golgi is responsible for adding mannose-6-phosphate to asparagine residues located on specific lysosomal enzymes. This mannose-6-phosphate is like the address label that tells the cell that those enzymes need to go to the lysosome. Again, lysosomes are where cells break down waste products and cellular debris. So the Golgi apparatus tags these enzymes with mannose-6-phosphate, which tells the rest of the cell that this is an enzyme for the lysosome. And one more time, which amino acids can be modified by the Golgi? Tyrosine, serine, threonine, and asparagine. That does get tested from time to time. Now, what happens if those lysosomal enzymes are not tagged with mannose-6-phosphate? Well, this causes something called I cell disease, or inclusion cell disease, which is an inherited lysosomal storage disorder. There's a deficiency in mannose phosphorylation, and without that mannose-6-phosphate address label to target the proteins to the lysosomes, these enzymes are secreted outside the cell. Now, this results in high plasma levels of lysosomal enzymes, clouded corneas, coarse facial features, and restricted joint movement. I cell disease is often fatal in early childhood, and it looks very similar to mucopolysaccharidoses such as Hurler syndrome or Hunter syndrome. Another important part of post-translational protein modification is protein folding. Polypeptides have to undergo folding into the proper three-dimensional conformation in order to become functional proteins. And there's a type of protein called a molecular chaperone that assists in this folding process. Chaperones assist in the proper folding and transport of polypeptides across the ER, Golgi apparatus, and beyond. So chaperones are important for the proper functioning of the ER. Some chaperones are synthesized constantly and are involved in the normal intracellular protein trafficking. Other chaperones are induced by stressors such as heat. So these are called heat shock proteins, HSP70 and HSP90. And these chaperones rescue shock stress proteins from misfolding. So if the folding process is not successful, the chaperones facilitate degradation of the damaged protein. This degradation process often involves ubiquitin, which is also a heat shock protein. Ubiquitin is added to the abnormal protein, which marks it for degradation by the proteasome. And we talked about how ubiquitin marks cyclins for degradation to help regulate the cell cycle. Well, ubiquitin also marks these abnormal proteins for degradation too. Now finally, let's talk about how we get rid of proteins that we don't need anymore. This is protein degradation, or proteolysis. Now, proteolysis can be accomplished by three different means. Option one is proteasomal degradation, which is where the protein is broken down by a structure called a proteasome. This process uses those ubiquitin tags again. So ubiquitin is attached to the defective proteins to mark those proteins for breakdown. Then the proteasome comes along and it contains an enzyme complex called ubiquitin protein ligase, and that recognizes the ubiquitin tag and then destroys the protein. Option two is lysosomal degradation, where the protein is sent to a lysosome and the lysosomal enzymes digest it. And we discussed that one already. And then option three for proteolysis is to use calcium-dependent enzymes that aren't found in the lysosome and aren't found in the proteasome, but again, they break down proteins. So there's a proteasomal mechanism which uses ubiquitin, there's a lysosomal mechanism, and there's a calcium-dependent enzyme mechanism. Three different ways to break down proteins that you don't need anymore. The next organelle I want to discuss is the sarcoplasmic reticulum. So this is a specialized kind of smooth endoplasmic reticulum that's found in muscle cells, both smooth muscle cells and skeletal muscle. So the sarcoplasmic reticulum stores calcium and it manipulates calcium balance in order to regulate muscle contraction. Calcium is one of the most important substances in the cell. Calcium is generally what makes things happen. Calcium allows muscle to contract. It triggers exocytosis of secretory products like neurotransmitters and hormones. So calcium balance within the cell is incredibly important, and the sarcoplasmic reticulum is an integral part of maintaining that proper calcium balance. Another incredibly important substance within the cell is ATP, which your cells need for energy. Now, ATP production takes place in the mitochondria through the TCA cycle and the electron transport chain, both of which take place within the mitochondria. So mitochondria are the powerhouses of the cell. Now, some cells are specialized in that they don't have very many mitochondria, less than 100. Other cells have more than 1,000 mitochondria. Mitochondria have an outer mitochondrial membrane, an intramembranous space, and then an inner mitochondrial membrane. And both of those membranes are lipid bilayers. Mitochondria are also interesting because they self-replicate, independent of what's going on in the nucleus, because mitochondria have their own DNA. No matter what DNA you got from your dad, you got your mitochondrial DNA from your mom, because all the mitochondria you had when you were a one-celled zygote came from your mom's ovum. So you can use mitochondrial DNA to trace groups of individuals that have a common maternal origin. And the mitochondria also play an important role in apoptosis. That'll be covered in Foundation 6. There's one other organelle I want to talk about, which is the peroxisome. Peroxisomes are involved in a lot of different metabolic processes, and I want to highlight 3 of those processes for you. First is the breakdown of very long-chain fatty acids through beta oxidation. Now, all the fats that you ingest are not the same. They're saturated fatty acids, monounsaturated fatty acids, polyunsaturated fatty acids, long-chain fatty acids, short-chain fatty acids, and so on. So the very long-chain fatty acids are broken down in the peroxisome by way of beta oxidation. The second important process is that the first steps in the synthesis of plasmalogens take place in the peroxisomes. Okay, great. What's a plasmalogen? Plasmalogens are a type of phospholipid that are found abundantly in myelin, in neural tissue. So I want you to bundle those terms together in your brain. Peroxisomes make plasmalogens, which are found in myelin. Then a third function of peroxisomes is that they contain oxidases and catalase, which are used to metabolize toxic substances that enter the cell, such as ethanol. So without peroxisomes in your hepatocytes, you would not be able to metabolize alcohol. So that's it for this video. Now we're ready for the end of session quiz. So pause the video, answer those quiz questions, and then we'll go through the answers. All right, first question. What must be present on a protein in order for that protein to gain entry into the nucleus? It's called a nuclear localization signal. Next, which cyclin CDK complexes assist in the progression from G1 phase to S phase? That was a complex of cyclin D plus CDK4 and also the complex of cyclin E plus CDK2. Next question, which cyclin CDK complexes assist in the progression from G2 phase to M phase? That was the complex of cyclin A plus CDK2 and the complex of cyclin B and CDK1. Next, what molecule does the Golgi apparatus add to proteins in order to direct those proteins to the lysosomes? It's mannose-6-phosphate. The last question, what are the different methods that a cell uses to break down proteins? So the 3 different methods we discussed for proteolysis are degradation by proteasomes, which is mediated by ubiquitin, degradation by lysosomes, and then degradation by those calcium-dependent enzymes. All right, that's the end of Foundations 3. I'll see you next time. We're still talking about cell biology, and we've already discussed the major organelles. So in this video, we're going to focus on the cytoskeleton. The cytoskeleton is the internal framework of a cell that gives it its specific shape and structure and orientation in space, depending on the function of the cell. There are several basic types of filaments that make up the cytoskeleton. There are microfilaments, which are the thinnest filaments. There are microtubules, which despite also being called micro, are actually the thickest type of filament. And then in between the microtubules and the microfilaments are the intermediate filaments, which have a diameter about half as large as the microtubules. And there's myosin, which is a little bit larger than the intermediate filaments. So again, going from the thinnest to the thickest, it's microfilaments, intermediate filaments, myosin, and microtubules. Now, microfilaments are made of polymers of a protein called actin. Actin is really important for cellular motility. Whether you're talking about neutrophil chemotaxis or the motility of macrophages, or even the microvilli of the intestinal brush border, or activated platelets that change shape to plug up a bleed. All those kinds of cellular movements are mediated by the growth and contraction of actin microfilaments. Now, myosin works right alongside actin to generate muscle contraction. So muscle cells have lots of myosin filaments. Remember, myosin, the myo means muscle. Myosin was first discovered in muscle cells, but there's actually an enormous family of myosin proteins, which are mostly involved in cellular motility in one form or another. And muscle contraction is certainly a big part of that. For intermediate filaments, take a look in your study guide. There are a whole bunch of different types of intermediate filaments, which are found in specific types of cells and tissues, and you can identify different tissues by staining for these different filaments. This really isn't all that clinically relevant unless you're going into pathology, but this is important for you to know for step 1. So the first one in your study guide is cytokeratin. Cytokeratin is a very strong filament found in epithelial cells. Cytokeratin contains the protein keratin, which is found abundantly in the skin. Cytoskeleton is found in desmosomes and also in hemidesmosomes, which are attachments between epithelial cells and other epithelial cells, and then between the epithelial cells and the basement membrane. That's going to be covered more in the dermatology videos. Now, we're going to come back and fill in the far right column about stains in just a second, but the next row is desmin. Now, desmin is found in all types of muscle cells, smooth muscle cells, skeletal muscle cells, even cardiac muscle cells. The next one is GFAP, or glial fibrillary acid proteins, which are found in glial cells, such as astrocytes and Schwann cells and so on. So this is a type of ectodermal intermediate filament. Then there are the neurofilaments, which can be categorized by their molecular weight. So there's low, medium, and high molecular weight neurofilaments. And they're found in neurons, as you might guess, but specifically they're found in the axons of the neurons. There are also nuclear lamins, A, B, and C. Now, these are definitely important for you to know for your exam. I mentioned previously that nuclear lamins are like the skeleton of the nucleus. They provide the structural framework for the nuclear envelope. And we talked about how the cyclin B CDK1 complex was associated with the breakdown of the nuclear envelope and the nuclear lamins, which has to take place during mitosis in order for the nucleus to divide. The other thing worth knowing about nuclear lamins is that the mutations in the nuclear lamin genes are implicated in several diseases, including certain types of muscular dystrophy and also the disease progeria. Progeria is a disease that causes advanced cellular aging, so that a 5-year-old child might have wrinkles in the facial structure of an 80-year-old because all these nuclear lamins are mutated. And then the last one here is vimentin. Vimentin is an important structural component of connective tissue, including fibroblasts and endothelium. It provides a strong support for cellular membranes, and it keeps organelles like the nucleus and the endoplasmic reticulum and the mitochondria fixed within the cytoplasm. Now let's go back and fill in the far right column of this table, and we'll talk about how different immunohistochemical stains can be used to identify tumors based on the intermediate filaments that they contain. Again, different tissues have unique intermediate filaments. So if you had a metastatic tumor somewhere in the brain or the liver or the lungs or wherever, you do a biopsy, and then the pathologist would stain that tissue to try to identify which kinds of intermediate filaments were present in the tumor. That's going to help you determine what kind of tissue the primary tumor came from. Sometimes it's hard to identify where metastatic tumors originated from, and these stains are going to help you identify what the origin of the tumor is. Cytoskeleton stain can be used to identify carcinoma, which is a cancer that arises from epithelial tissue. Desmin is found in muscle cells, we said. So if you have a sarcoma of the muscle cells, a myosarcoma, you could use a desmin stain to help you identify that tumor. So this might be like a rhabdomyosarcoma or a leiomyosarcoma. GFAP is a helpful stain in identifying certain types of astrocytomas, such as a glioblastoma. And then a neurofilament stain stains neurons rather than glia. Now this can be a useful stain to help you identify a tumor of neural origin, such as a neuroblastoma, which is a childhood tumor that arises from sympathetic ganglion cells in the adrenal medulla. And then again, vimentin is found in connective tissue. So that stain is mainly helpful in identifying sarcomas and also some carcinomas. But primarily I want you to associate vimentin with sarcoma, and you can associate cytoskeleton with carcinomas. So hopefully these aren't too hard, but chances are you're just going to have to sit down and memorize these. The largest type of cytoskeletal filament are the microtubules. Microtubules are tubes made up of proteins called tubulin. And there are two types of tubulin, alpha tubulin and beta tubulin. And first they come together to form a dimer, and then the dimers join together lengthwise to form something called a protofilament. And then those protofilaments come together side by side to form a cylindrical microtubule with a hollow core. And the way the alpha and beta subunits are arranged, they form a spiral or a helix that runs the length of the microtubule. Now in terms of function, I want you to think movement. Microtubules are involved with movement, a couple of different kinds of movement. For starters, microtubules are an important part of the structure of flagella and cilia, and they're what enable the cilia to move, as we're going to see shortly. Microtubules are also part of the mitotic spindles, which are what pulls the duplicated chromosomes apart during nuclear division. Now, this is a simplified explanation, but basically during mitosis, the microtubules assemble toward the chromosomes and latch on, then they disassemble very rapidly and pull those chromosomes apart so the cell can divide. Microtubules polymerize and grow very slowly, but they can collapse fairly quickly. So if something prevents the microtubules from growing and then collapsing, that'll interrupt mitosis. So what drugs act on microtubules? Well, the first group are cancer drugs, and there are two groups of cancer drugs that act on microtubules: vinca alkaloids and taxanes. The Vinc alkaloids are vincristine and vinblastine, and they work by blocking the polymerization of microtubules. They prevent the microtubules from growing. And we're going to talk about that at the very end of the course in the oncology section. And then the taxanes, or taxols, include drugs like paclitaxel and docetaxel, which work by hyperstabilizing the microtubules. So the microtubules can grow, but then they can't break down in order to pull those chromosomes apart. So the vincas and the taxols are similar in that they both interfere with microtubule function and they prevent mitosis in cancer cells, but on a cellular level, they actually work in very different ways. Then, other than the cancer drugs, there are the benzimidazoles, which are antihelminthic drugs like mebendazole, albendazole, thiabendazole. These are used to treat certain worm infections, hookworm, tapeworm, that kind of thing. You can remember that worms are bendy, so you're going to use mebendazole and albendazole to kill them. Griseofulvin is an antifungal drug that works on microtubules. It's mostly used for ringworm and nail fungus. There's also colchicine, which is an anti-inflammatory drug used to treat gout. So phagocytes use microtubules to move, and colchicine helps to reduce inflammation because the phagocytes can't move to where they need to go because they're using those microtubules to move and the drug is interfering with the microtubules. Now, one other function of microtubules is to allow the cell to move things around within the cytoplasm. Remember, microtubules are involved with movement. So they help cilia move, they help phagocytes move, they help chromosomes move during mitosis, and now they're helping the cell actually sort of ship things around from one part of the cell to the other. For instance, some cells are really big. Take a motor neuron for your big toe. The cell body is way up in the anterior horn cells of the L4, L5 region of the spinal cord, but the axon stretches all the way down to the big toe. That could be 3 or 4 feet long, depending on how tall you are. So there needs to be a mechanism to move things reliably from the cell body to the other end of the cell. So this process is called slow axoplasmic transport, and it basically uses the microtubules like a railroad track to guide things to the right location inside the cell. It's not just neurons. Other cells use microtubule tracks to move things around. So how exactly does this work? Well, the railroad cars that actually move things up and down these microtubule tracks are called molecular motor proteins. And there are 2 of these molecular motor proteins that you should know about, dynein and kinesin. Microtubules are oriented with a plus end and a minus end. The end of the microtubule made up of alpha tubulin is the minus end, and the end with beta tubulins is the plus end. So dynein and kinesin attach to the outside of the microtubule, and they also attach to whatever cargo they're hauling, and then they walk along the microtubule from one end to the other, dragging their cargo with them. Kinesin walks toward the plus end of the microtubule, which is generally going to be away from the nucleus, so that's called anterograde transport. And then dynein walks in the other direction, toward the minus end, which is called retrograde transport. We're going to come back to dynein in just a second. Let's also talk about cilia and flagella, because they're both composed of microtubules. The flagella of sperm are structurally very similar to a cilium, just a lot longer. In motile cilia and flagella, the microtubules come together in a 9+2 arrangement. A circle of 9 pairs, or doublets, of microtubules on the outside, and then 2 individual microtubules running down the center. And then lots of dynein motor proteins stick out from the microtubules and walk along adjacent doublets, which allows each microtubule doublet to move relative to another doublet, so that the cilium moves as a unit. Now, where in the body do we find cilia? We find them in the respiratory tract, including the trachea and the bronchioles, and also the paranasal sinuses, because cilia help to sweep out mucus and bacteria and debris. The fallopian tubes are ciliated because the cilia help to sweep the ovum, or a fertilized zygote, down toward the uterus. So just keep that list of ciliated structures in your mind for just a moment. Then one other thing to know about how ciliated cells work is how they coordinate their ciliary motions. If each ciliated cell was doing its own thing, you wouldn't really be able to sweep anything along in a particular direction. You'd just kind of be going randomly. But instead, what we actually see is a coordinated ciliary contraction taking place that moves things in one direction. So how does one ciliated cell communicate with another ciliated cell to coordinate the contraction and create a wave-like motion? Well, it's accomplished by gap junctions between the cells. So gap junctions are important for the coordinated contraction of ciliated cells. That's just a tiny detail that's worth knowing for your test. Now, what happens if the molecular motor proteins in the cilia don't work? Well, there's a condition called primary ciliary dyskinesia, where the dynein doesn't work. and that leads to a lack of movement of the cilia in the respiratory tree, and in the sinuses, and in the fallopian tube, and even in the flagellum of the sperm. So in primary ciliary dyskinesia, there are immotile cilia due to a defect in the dynean arms. If the dynean can't walk like it's supposed to, you're not going to have movement of the cilia. So this results in infertility in both males and females, because in the males, the sperm are immotile, and in females, because the fallopian tubes aren't going to be able to sweep the zygote or the oocyte anywhere. There's also bronchiectasis, because these patients can't clear mucus out of the bronchial tree. There's chronic sinusitis, because they can't clear out the sinuses. And then cilia are also involved in determining the position of internal organs during embryological development. So about 50% of patients with primary ciliary dyskinesia also have situs inversus, where the organs are on the opposite side of the body. So the liver would be on the left and the spleen would be on the right. There's also dextrocardia, where the heart is over on the right. Now, not all patients with situs inversus have primary ciliary dyskinesia, and not all patients with primary ciliary dyskinesia have situs inversus. Now, if you have the triad of situs inversus, bronchiectasis, and chronic sinusitis, that's called Cartaginer syndrome. But again, about 50% of patients who have primary ciliary dyskinesia do not have situs inversus. So Cartaginer syndrome does not equal primary ciliary dyskinesia. When the cilia don't work, there's a 50-50 chance of whether your organs are going to be on the right side or the wrong side. So that's it for this video. Go ahead and answer the questions in the end of session quiz, and then we'll go over the answers together. All right, first question. What drugs act on microtubules? Well, I mentioned 4 cancer drugs: vincristine, vinblastine, paclitaxel, and docetaxel. Then there are a few others: benzimidazole antihelminthic drugs, mebendazole, albendazole, and thiabendazole. There's griseofulvin, which is an antifungal, and there's colchicine, which is an anti-inflammatory that acts on the microtubules in phagocytes. Next, what are the intermediate filaments found in the following tissue types and cellular structures? So in connective tissue, you see vimentin. In muscle tissue, you see desmin. In epithelial tissue, you see cytokeratin. And in the axons, you find the neurofilaments. And the last question, what are the defects seen in Cartaginer syndrome? So Cartaginer syndrome is a triad of bronchiectasis, chronic sinusitis, and situs inversus. And then because Cartaginer syndrome can also be a form of primary ciliary dyskinesia, you can also add infertility to that list, because infertility is a major feature of that disorder. So that's it for Foundations 4. I'll see you next time.
Two drugs with mechanisms not the same. The vincas and the taxanes are their names. One inhibits, the other a microtubule enhancer. Yet both are used to fight the dreaded cancer. As our tragic story doth begin, starring these inhibitors of tubulin, a pair of star-crossed lovers form our scene. Brave paclitaxel and his vincristine. Soft! What light through yonder window breaks? It is the east, and vincristine is the sun. Arise, fair vinca alkaloid, my sweet. Oh, that I were a Ewing sarcoma, and that thou wouldst inhibit polymerization of my microtubules and disrupt my mitotic spindles.
Oh, paclitaxel, paclitaxel, wherefore art thou paclitaxel? Oh, what's in a name? That which we call a taxane by any other name would smell as sweet.
Why must our families oppose one another this way?
It is our very nature, my love. We vincas inhibit cell division by inhibiting microtubules, while you taxanes stabilize microtubules.
Yes, but the end result is the same. Oh, why must this keep us apart?
Besides, you taxanes cause me such devastating hypersensitivity reactions.
Premedicate! Premedicate, my queen! With a steroid and an antihistamine!
Ronnie, this isn't working.
No, you're doing great! This show must go on!
This is a terrible way to study. I don't have time for these stupid role-playing games of yours. Can't believe I put on a corset for this.
In this video, we're going to talk about the plasma membrane. and how the plasma membrane interacts with both the cell contents and the cell's external environment. Let's start off talking about the composition of the plasma membrane. The plasma membrane is a double layer of phospholipid molecules with the polar hydrophilic head oriented both on the outer surface of the cell and also inside the cell, and then the hydrophobic lipid tails are oriented inward. So this is a phospholipid bilayer. And I think it's worth mentioning that there are many different types of phospholipids, including phosphatidylcholine, which is also known as lecithin, phosphatidylserine, and there's phosphatidylinositol. Now, that phospholipid bilayer is the basic structure of the plasma membrane, but it's not the whole story. The plasma membrane is a very complex structure, and it contains a lot of other things, such as a huge variety of proteins, such as transport proteins, tyrosine kinase receptors, G protein-linked receptors, and so on. By mass, there's actually more protein in the plasma membrane than there are phospholipids. You also have glycoproteins, glycolipids, sphingolipids like sphingomyelin, and cholesterol, which makes up about 15% of the plasma membrane in our cells. And cholesterol is a more rigid structure than phospholipids, so including cholesterol in the plasma membrane prevents the membrane from being too fluid. So a little bit of cholesterol stabilizes the plasma membrane, but if there's too much cholesterol in the membrane becomes too rigid. And additional cholesterol also increases the membrane's melting temperature. Now, one of the things that makes each cell type unique is the unique type and number of receptors that are incorporated into the cell's plasma membrane. And there are three main families of receptors that we're going to focus on throughout Part 2 of the course. There are tyrosine kinase receptors, G protein-linked receptors, and steroid receptors. Now, the first two types, the tyrosine kinase receptors and G protein-coupled receptors, are transmembrane receptors. They're part of the plasma membrane, and they're kind of embedded in the lipid bilayer, and that's where they perform their functions. But steroid receptors are found inside the cell, kind of floating around in the cytoplasm. Steroid hormones like estrogen or testosterone are able to pass through the plasma membrane, and then they bind to their receptors inside the cell itself. But tyrosine kinase receptors and the G protein receptors are located on the plasma membrane. Now, we're going to talk about the G protein receptors in detail in the Pharm Basics section of the course. But for now, let's focus on the tyrosine kinase receptors. Take a look in your study guide. Tyrosine kinase receptors are transmembrane receptors that bind to some extracellular ligand, and then on the part of the receptor that's inside the cell, they transfer phosphate groups to selected tyrosine side chains on specific cellular proteins. That's called phosphorylation. So a kinase is an enzyme that phosphorylates things. It just means it adds phosphate groups to things. And a tyrosine kinase only phosphorylates tyrosine residues. And the very first step in this signaling cascade is autophosphorylation, which means that the tyrosine kinase phosphorylates itself. We're going to come back to that in a moment. Some tyrosine kinase receptors, like the receptor for platelet-derived growth factor, are single-pass transmembrane proteins. That means the receptor protein doesn't pass back and forth through the plasma membrane several times. It just goes through one time. It's a single-pass receptor. But other types of tyrosine kinase receptors, like insulin receptors and insulin-like growth factor receptors, have multiple subunits. So it's not just a single pass. For instance, the insulin receptor has 2 alpha subunits and 2 beta subunits. Those subunits are all held together by disulfide bonds, which are very strong bonds. If you need to hold something together very tightly, you're going to use a disulfide bond. We often find disulfide bonds outside the cell in the extracellular environment where conditions are harsh, so you need a really strong bond to hold something together. So a molecule of insulin binds to the 2 alpha subunits, which are on the outside of the cell, and the 2 beta subunits that have a tyrosine kinase activity are actually found inside the cell. And when insulin binds to the alpha subunits, that causes a conformational change in the receptor, which allows the beta subunits to autophosphorylate. And that in turn allows the tyrosine kinase domain to start phosphorylating other proteins to have some downstream effect. That's it for tyrosine kinases, but let's quickly review some basic enzyme terminology, which is going to come up again and again. Again, an enzyme that uses ATP to add phosphate groups to something is called a kinase. An enzyme that removes a phosphate group is called a phosphatase. An enzyme that adds inorganic phosphate to something without using ATP is a phosphorylase. An enzyme that adds a carboxyl group is a carboxylase. And this usually requires biotin or vitamin B7 as a cofactor. An enzyme that removes a carboxyl group is a decarboxylase. Likewise, an enzyme that adds a hydroxyl group is a hydroxylase, and the enzyme that removes a hydroxyl group is a dehydroxylase. But I don't want you to get dehydroxylase confused with dehydrogenase. A dehydrogenase is an enzyme that oxidizes something by using an electron acceptor, such as NAD or NADP. So be sure you're familiar with all that terminology. One key function of the plasma membrane is to keep potassium inside the cell and to keep sodium outside the cell. Our cells use this sodium gradient to drive the active transport of a lot of different substances into the cell. Sodium naturally wants to enter the cell going down its concentration gradient, and the cell can use that energy to drive the transport of other things against their concentration gradients. So it's important for cells to be able to constantly fine-tune the sodium content within the cell. And potassium is also important because high potassium content inside the cell keeps the cell's resting membrane potential around -55, which is important for neurotransmission and muscle function and so on. So, how do cells manage to maintain the sodium and potassium gradients? You know this, it's the sodium-potassium ATPase. This pushes out 3 sodium ions for every 2 potassium ions that come in. And because sodium is constantly trickling back in and potassium is constantly trickling out, this sodium-potassium ATPase has to work continuously. And because it's an ATPase, you know that this process requires energy in order to maintain this gradient. You also need to know that the drug digoxin, or digitalis, is a cardiac glycoside that works by inhibiting the sodium-potassium ATPase. When we get to the cardiovascular section, we'll explain how inhibiting this enzyme will eventually increase intracellular calcium, and that makes the heart beat a little harder. Not everything that enters the cell enters through a protein channel. Lots of macromolecules enter the cell through endocytosis, which also requires some specialized equipment. So let's talk about receptor-mediated endocytosis for a minute. The molecule I want you to focus on is clathrin. Clathrin is a vesicular trafficking protein, similar to the coat proteins that we discussed with the Golgi apparatus. And clathrin helps the plasma membrane form spherical vesicles during endocytosis. Let's take a look at your study guide. Instead of binding to the endoplasmic reticulum or the Golgi apparatus like the coat proteins, clathrin binds to a protein called adaptin on the inner surface of the plasma membrane, and that adaptin is bound to a receptor protein. That receptor is going to bind some specific cargo that needs to be transported into the cell by endocytosis. It might be an LDL particle or transferrin or any number of things. Once that cargo molecule binds to the receptor, the clathrin molecules start to polymerize with other clathrin molecules. And just like we saw with the coat proteins, clathrin polymerizes into little spheres. So this allows the plasma membrane to fold up into a little vesicle. Then another protein called dynamin pinches that off and helps the two ends of the membrane fuse. Then you have formed a little vesicle that's coated with clathrin. And finally, the clathrin and adaptin can come off the vesicle and be reused, so you're left with a naked transport vesicle with just receptors and the cargo molecules inside. Then something needs to happen to the contents of that vesicle before they can be released inside the cell. Let's look at the next drawing. What typically happens after the uncoating is that the receptors get recycled back to the plasma membrane, and what's left of the vesicle fuses with a lysosome so that all those hydrolytic enzymes inside the lysosome can break down whatever was brought into the cell. Now in this example, it's an LDL particle, so the lysosomal enzymes are degrading the lipoproteins, and then the cholesterol is released into the cytoplasm. So this is the process of receptor-mediated endocytosis. Now let's look at this bottom image as an example of how this is clinically relevant. On figure A, you have the normal LDL receptor and adaptin and clathrin. And you can see that these come together in what we call clathrin-coated pits. But on figure B, there's a mutant LDL receptor that doesn't associate with adaptin and clathrin like it's supposed to. So these individuals can't bring the LDL into the cell and use it. So this is what you might see with familial hypercholesterolemia, where there's a defective LDL receptor. But we're going to talk more about that disease in another video. So that's clathrin. It's definitely an important mechanism to know about. Finally, let's talk about arachidonic acid derivatives, which are ultimately derived from plasma membrane phospholipids. Now, this is very important for you to know for your test, and it's very clinically relevant to know about how certain drugs interact with this arachidonic acid pathway. This is a 4-star topic, so don't breeze through this. Fundamentally, arachidonic acid derivatives are substances that allow for tissue-to-tissue communication. Things like leukotrienes and prostacyclin and prostaglandins are all communicating substances, and they're all derived from arachidonic acid. Let's look at this pathway in your study guide, and you can fill it out as we go along. You start out with cell membrane phospholipids, specifically phosphatidylinositol. Now, phosphatidylinositol is then converted to arachidonic acid, which is letter A. Now, enzyme B is the enzyme that catalyzes this reaction. So B is phospholipase A2. That's an important enzyme to know. Phospholipase A2 is inhibited by glucocorticoids, which is letter C. So glucocorticoids have a lot of functions, and one of the things they do is inhibit this enzyme, and therefore inhibit the production of arachidonic acid. Then arachidonic acid can go down one of two pathways. First, arachidonic acid can be acted upon by letter D in your diagram, which is 5-lipoxygenase, to form hydroperoxides, which is letter F. Specifically, it forms 5-HPETE, or 5-hydroperoxyeicosatetraenoic acid. Don't worry about that long name. Just remember 5-HPETE. And there's a drug that inhibits 5-lipoxygenase, which is xyluton, letter E. Now, xyluton is a drug that's used occasionally to treat asthma. So 5-HPETE is converted to leukotrienes, which is letter G. There's leukotriene B4, which we're going to mention again in the immunology section, but it's associated with neutrophil chemotaxis. So if you want to get neutrophils to an area, then you're going to convert arachidonic acid to leukotriene B4. That's going to tell the neutrophils, hey, come over here. And then other leukotrienes are involved in increasing bronchial tone, which is why blocking leukotrienes is helpful in asthma patients. So letter H is neutrophil chemotaxis and increased bronchial tone. Then letter I is two more drugs, zafirlukast and montelukast. They both have Lu in the name because they block leukotriene receptors. And again, they interrupt that increased bronchial tone. So these are useful in the treatment of asthma. So xyluton inhibits the formation of leukotrienes, and zafirlukast and montelukast block the leukotriene receptors. And then you may want to know that montelukast is approved for younger age groups. So if you have, say, a 2-year-old with asthma, you're going to start on a leukotriene inhibitor, you should choose montelukast. That's something you need to know for your test. We're going to talk about that again in the pulmonary section. Now let's go back up to arachidonic acid, letter A, and look at the other side of this pathway. Arachidonic acid can also be acted upon by letter J, which is the enzyme cyclooxygenase, either COX-1 or COX-2. And letter K is a bunch of different drugs that inhibit cyclooxygenase, including NSAIDs, acetaminophen, celecoxib, which is a COX-2 inhibitor, and aspirin. Now, aspirin irreversibly inhibits cyclooxygenase. And it's worth mentioning here that although acetaminophen inhibits cyclooxygenase, it's inactivated peripherally, so it has no antiplatelet effects or anti-inflammatory effects. And then letter L is endoperoxides, which are the products of cyclooxygenase. Now from there, the endoperoxides can go down 3 different pathways. Letter M is prostacyclin, or PGI2, which stands for prostaglandin I2. It may help you to think of PGI as being platelet gathering inhibitor, because PGI2 decreases platelet aggregation. It also decreases vascular smooth muscle tone and uterine muscle tone. So it's going to abolish uterine contractions. So box N is decreased platelet aggregation, decreased vascular tone, and decreased uterine tone. And as a vasodilator, analogs of prostacyclin are used to treat conditions like Raynaud phenomenon and pulmonary hypertension. Then letter O is prostaglandins. So PGE2 and PGF2 alpha increase uterine tone, so they're going to be helpful to either induce labor. They're also useful as abortifacients because they induce uterine contractions. And PGE1 decreases vascular tone. So again, box P is increased uterine tone and decreased vascular tone. And then the third pathway is to convert the endoperoxides to thromboxane A2, which is letter Q. Now thromboxane A2 is prothrombotic, as you might have guessed from the name. So thromboxane A2 increases platelet aggregation. It also increases vascular tone. So that's box R, increased platelet aggregation and increased vascular tone. It's just the opposite of PGI2. So it may be helpful to think about thromboxane A2 and PGI2 as having opposite effects. So again, you should definitely spend some time with this pathway. Make sure you really know it, because you will see these substances again throughout the course and on your test. So that's it for the plasma membrane. Go ahead and work through the end of session quiz, and then we'll go over the answers together. All right, first question. Which arachidonic acid product causes each of the following effects? So increased bronchial tone are going to be leukotriene C4, leukotriene D4, and leukotriene E4. Increased platelet aggregation is thromboxane A2. Decreased platelet aggregation is prostacyclin, or PGI2. Increased uterine tone is prostaglandins E2 and F2 alpha. Decreased uterine tone is prostacyclin, PGI2. Increased vascular tone is thromboxane A2. And decreased vascular tone is prostaglandin E1 and also prostacyclin, or PGI2. Next, what are the two most abundant substances in the plasma membranes? Well, it's proteins and phospholipids. And the last question, what drugs act on the arachidonic acid product pathway? And what enzymes do they affect? Glucocorticoids inhibit phospholipase A2. Xyluton inhibits leukotriene production by inhibiting the enzyme 5-lipoxygenase. Zafirlukast and montelukast inhibit the leukotriene receptors. And then the enzyme cyclooxygenase is inhibited by NSAIDs, acetaminophen, COX-2 inhibitors, and aspirin. All right, that's it for now. I'll see you next time.
Hello everyone, I'm Dr. Chris Lewis, and this is my first video of the course. You've already watched a few videos and might be asking yourself, what can you offer me, Dr. Lewis? Well, I'll tell you. Do I have mnemonics? Yes. Do I have high-yield teaching? You betcha. Do I have pathology slides? Heck yeah, I do. And that's not all. I have a bunch of hats that I might wear. Well, not this one. This one's dumb. And I've got these super cool pink future glasses and these highlighters, which may not be much use for you directly, but they're still pretty awesome. So strap yourself in, because with all this stuff, you and I are gonna kick some step 1 butt. Ah! No, more explosions. Well, that was weird. Hello everyone, and welcome to our Step 1 video on cellular suffering and death. Oh, it sounds so sad, but exciting. So first let's talk about apoptosis. So apoptosis is an orderly cell death process. And when we say orderly, we mean that we expect and actually plan for this cell death. Now it is a natural and needed process that serves to get rid of cells that are no longer of any use. So why is apoptosis so important? Why do we need a complicated process to allow our cells to die? That should be easy, right? Well, if we let cells die in an unorganized manner, all these cell contents would spill out and would then produce an inflammatory response, which is not ideal. So we have developed a process by which these doomed cells are broken up into small kind of bite-sized pieces, which are then phagocytosed by other cells with no messy debris floating about, and most importantly, no inflammation. So, what causes apoptosis? Well, there are physiologic situations and pathologic conditions. So the physiologic causes will include things like during embryogenesis, so we don't all have 12 fingers or a bunch of tails. Cell breakdown during menstruation is another example. And there's also proliferation cell populations in the body, like immature lymphocytes that we need to get rid of and go through that programmed cell death in order to maintain a constant cell number. And then there are pathologic causes, DNA damage to a cell through radiation or hypoxia, misfolded proteins that occur due to gene mutations or exposure to free radicals. Infections like HIV will induce cells to undergo apoptosis. So you need to know the descriptive histological characteristics of apoptosis. So what are you going to see here? Well, first, most cells are going to shrink. They're going to get smaller. Also, something called pycnosis occurs. Well, what's that? Well, pycnosis is condensation of the nuclear chromatin, which results in shrinkage of the nucleus as well. And this causes the cell to look basophilic as well. You'll also see some membrane blebbing, something called karyoresis. That's nuclear fragmentation. And the formation of apoptotic bodies. So things are breaking apart into smaller pieces, which are then phagocytosed. Now, another characteristic of apoptosis is DNA laddering, which can be seen on agarose gel electrophoresis. So capsase activated DNase will cleave DNA into fragments that are about 180 to 185 base pairs in length. So the pattern you see on the electrophoresis kind of looks like a ladder. Again, no inflammation is taking place with apoptosis. You're not going to see that red, hot, warm area that's very classic of an inflammatory process. So with apoptosis, you have the cytoplasm blebbing away, and it's taken care of by phagocytes. Now, the cell is shrinking bit by bit, and again, there is no inflammation. So now we get to the tough part of apoptosis, the mechanism. Now before we get too far into the specific pathways of apoptosis, I want you to know that the ultimate initiators of apoptosis are a family of proteins called caspases. Now they exist in an inactive form in cells, and then when they're cleaved, they become active, and then they carry out many important functions associated with initiating apoptosis. So that's just the end of the story. Caspases equal apoptosis. Now, I wish that was all you needed to know. Let's go back to the beginning of the story and talk about the pathways. Now, the two pathways are the extrinsic and the intrinsic pathway. First, let's talk about the intrinsic pathway. So this is the major mechanism of apoptosis, and it's used for both the physiologic and the pathologic conditions that are going to lead to this. It's also referred to as the mitochondrial pathway because it involves an increase in mitochondrial permeability, which leads to the release of pro-apoptotic molecules. So let's get into this. So BCL2 is a family of proteins that closely regulate the permeability of the mitochondrial membrane. Now, mostly they act as an anti-apoptotic protein. Now, if there is DNA damage or another apoptotic signal is received, then another family of proteins called the BAX or the BAX will go into the mitochondrial membrane and create channels that allow the contents of the mitochondria to flow out. So let's review. BCL2, anti-apoptotic. BAX or BAX is pro-apoptotic. Now, one way to remember this is that BCL2 has 4 letters and numbers, and live or live has 4 letters. BAX has 3 letters, and then also die has 3 letters as well. And BAX also rhymes with axe, so BAX is giving axe to cells. That's the way I remember it. And again, that BAX is letting the contents of the mitochondria out. So what's in the mitochondria that's so bad for cells? Well, the answer is cytochrome C. Cytochrome C will then start a cascade of enzyme activations that will end up activating caspases, which we talked about earlier before, and that again is going to lead to apoptosis. So next, let's talk about the extrinsic pathway. So you can remember this because the signal is coming from outside of the cell. And there's actually kind of 2 different mechanisms here. First, there are the death signals. In the cell membrane, there are 2 different receptors that can receive those death signals. And this includes the TNF receptor and the FAS receptor, also referred to as the CD95. So they will be activated by either TNF alpha or by the Fas ligand, respectively. Now, once activated, the receptors will transmit signals that will again cause activation of caspases. Now, another extrinsic mechanism occurs when killer lymphocytes, mainly cytotoxic T cells, recognize a cell as foreign or infected. So these lymphocytes will then release perforin and granzyme B. So what do? Well, perforin will punch holes in the plasma membrane. Remember, perforin will perforate. That's the way I remember that one. And then that granzyme B is going to enter into the cell and activate the caspases directly. All right, so let's go over this again in a Lewis Notes. Hello everyone, welcome to our very first Lewis Notes, and Lewis Notes are what we're going to use some throughout this course in order to really knock home some of our highest yield stuff. So what I like people to do is break out their own piece of paper and write in some of this stuff yourself, because the more you put it into your brain, especially if you're writing it, the more likely that you are to retain it. So we're going to do a matching game, and you'll see me do a lot of matching games throughout the course. So here we're looking at our intrinsic and extrinsic, and we boiled it down to the most important stuff, and we're going to do a matching game here. So let's start with the intrinsic pathway here. So first we have the anti-apoptotic stuff. Remember, anti has 4 letters in it, so that's going to be our BCL2, and we're going to put that right over there. What about our pro-apoptotic stuff? Remember, that's going to be our 3-letter one, so that's going to be BAX. So if something activates BAX, then you're going to start this whole cascade down. So if you get BAX that's stimulated, you're going to get an increase in what? Remember, this is going to be the increase in mitochondrial permeability. And you don't want to do that, because once you get mitochondrial permeability up, you're going to start spewing cytochrome C all over the place, and that's going to lead to further initiation of apoptosis. Now let's move over here to the extrinsic pathway, and first we have our death receptors. That sounds like a cool band name. So our death receptors, the first one we have here is the TNF receptor, and if that gets stimulated, then you're going to run into problems. The other one that you need to remember is the FAS receptor, also known as CD95. Now, the other way that the extrinsic pathway can work is through killer T cells, so those cytotoxic T cells. And it's going to use things like perforin. Remember, you're going to use perforin here, and that's going to put holes in the plasma membrane. And the plasma membrane is wide open for granzyme B to sneak on in. That granzyme B is going to go enter in the cell, and it's going to lead to activation of caspases. And then those caspases are going to then promote apoptosis. So if you can remember all that stuff right there, then you're going to be good to go on the intrinsic and the extrinsic pathway. Now, one last mechanism of apoptosis that maybe doesn't fit in either pathway is protein P53. So P53 is a very important player in DNA repair. Now, if DNA damage is present, P53 will arrest the cell in stage G1 of the cell cycle. So this will actually allow time, it's just stalling, for the DNA to be repaired by repair enzymes. Now, if the damage is irreparable, then P53 will then activate mechanisms that will then again lead to apoptosis. Now, P53 is often referred to as a tumor suppressor protein because if P53 is dysfunctional, then this can lead to tumor formation in cancer. All right, so now let's move on to necrosis. So necrosis is cell death that is not orderly. So everything we try to avoid in apoptosis tends to occur in necrosis. Now, all the intracellular components are spilling out into the world, and as you would expect, this causes inflammation. Now, there are several types of necrosis that you should be aware of. Now, most of the descriptive terms refer to their microscopic appearance, but we also see this necrosis and these types of necrosis in specific organ groups as well. So the first one is coagulative, and coagulative is characterized as kind of a gelatinous substance in dead tissue, and it occurs in the heart, in the liver, and the kidney, and it occurs in low oxygen environments, so maybe after an infarction. Next, we have liquefactive. So this is a viscous liquid mass that occurs more in the brain and very often and commonly in bacterial abscesses and pleural effusions. Infections cause an inflammatory response that is going to lead to this type of reaction. Next, we have caseous necrosis. So this is a kind of a combination of coagulative and liquefactive, and it tends to look like kind of clumpy cheese. It's really nasty. So this is seen very commonly in tuberculosis, systemic fungal infections as well. Next, we have fatty or fat necrosis. This results from the activation of lipases and can be seen with pancreatitis, can also be seen in subcutaneous tissues as well. Next, we have fibrinoid necrosis. So this is in blood vessels, so think vascular things. So this is caused by autoimmune or just immune-mediated vascular damage. And then we have gangrenous necrosis. That can be either wet or dry. We'll go into great detail of that in a future lecture. The wet version is seen with bacterial infections. It's common in extremes. I always think of like Civil War injuries and having to cut off limbs with saws with that type of gangrene. There's also a dry version, and this is seen with ischemia, and it occurs in the toes and the feet. And it's mainly due to arterial occlusion. You'll see those really nasty, like black toes. All right, so we've established that necrosis is messy and cellular contents are spilling into the body. If you suspect pathology in certain organs or cells, then you can test for these contents or markers to see if cell death is occurring. So we're going to look into the study guide here. What cellular byproducts might you detect in the serum when the following cell types are injured? So let's talk about cardiac first. So cardiac myocytes, when they die, what are you going to get? We're going to get things like myoglobin. You're going to get CK, CKMB. You're going to get troponin I. So when someone comes in the ER with chest pain, we always check for cardiac enzymes, especially troponin I and CKMB. Now, what about with skeletal myocytes? What if they die? What are you going to see? Well, you're going to see an increase in CK, aldolase, again, myoglobin. In patients with rhabdomyolysis, muscle breakdown, is going to release massive amounts of myoglobin, which is actually going to be toxic to the kidneys, so you can end up with renal failure. What about hepatocytes in the liver? If you have a lot of liver damage, you're going to see an increase in AST and ALT, alkaline phosphatase, and GGT. If you have damage to salivary glands, you're going to see an increase in amylase. If you have problems with the pancreas, you're going to see increases in amylase and lipase. If you have RBCs or red blood cell damage, you're going to see heme, which will then break down into bilirubin, and that'll be elevated. All right, so let's talk a little bit more about cell injury. It's important to know the different characteristics of cell injury and what those characteristics of reversible cell injury or an irreversible cell injury. So with reversible cell injury, which is usually correctable with sufficient oxygen and nutrients, this is characterized by things like decreased ATP synthesis, cellular swelling, nuclear chromatin clumping. But be aware that sometimes that chromatin clumping can also be seen with irreversible injury as well. You can also see this with a decreased glycogen, a fatty change of the liver or of the cells. So you might see this in hepatocytes if you go maybe binge drinking or if you have a Tylenol overdose. Also ribosomal detachment resulting in decreased protein synthesis. Now, the characteristics of irreversible cell injury, injury that your cells can't come back from, are things like the nuclear pycnosis, the irreversible condensation of the chromatin. You can see karyolysis, the complete dissolution of the chromatin. Karyoresis, which is the fragmentation of the nucleus. You can also see this with calcium influx causing caspase activation, plasma membrane damage, lysosomal rupture, increasing mitochondrial permeability. Again, all irreversible cell injuries. And then next, I want to cover briefly some of the types of cell injury. What are the major culprits when it comes to damaging our cells? So the first one is ATP depletion. So without ATP, really nothing works. The major causes of ATP depletion are reduced supply of oxygen and nutrients, mitochondrial damage as well, and actions of even some toxins like cyanide. What else can cause our problems? Mitochondrial damage. We talked about this a little bit before. So this is closely related to our first mechanism because mitochondria are the suppliers of our ATP. Ultimately, this will impair ATP production. Now, the mitochondria have an outer and an inner membrane, and that houses several proteins that are capable of activating apoptotic pathways. So remember, we already talked about how those cytochrome Cs can activate caspases in the cytosol. Therefore, if you damage the mitochondria, then you might signal apoptosis and that programmed cell death. Now, mitochondria can be damaged by increases in cytosolic calcium, by reactive oxygen species, and oxygen deprivation. Also, mutations in mitochondrial genes are a cause of some inherited diseases as well. Let's talk about an influx of calcium and a loss of calcium homeostasis. So ischemia and certain toxins cause an increase in cytosolic calcium concentration. So this increased calcium can result in an increase of the mitochondrial permeability transition pore and thus impair ATP production. Now, increased calcium can also activate certain enzymes like phospholipases, proteases, endonucleases, and ATPases, which all can result in negative cellular effects. Next, let's talk about accumulation of oxygen-derived free radicals, so also referred to as maybe oxidative stress. So free radicals cause cell damage through membrane lipid peroxidation, through protein modification and DNA breakage, free radical injury. They can also created by a lot of different things. So how do you get those free radicals? Well, radiation exposure, metabolism of drugs, redox reaction, nitric oxide, transition metals, a leukocyte oxidative burst. Now, some of these are desirable, like the leukocytes creating an oxidative burst, because this can be useful in killing bacteria. Of course, radiation would not be a good way to encounter free radicals. Reperfusion after anoxia is another problem. That's going to produce a free radical production. So this is actually a major cause of injury after thrombolytic therapy. So this is why we have windows for thrombolytic therapy. So say in a stroke patient, if you can catch the ischemic stroke within maybe 3 hours, you can go ahead and give your thrombolytic. If it's beyond 3 hours or even beyond 6 hours, you can't do it because of that reperfusion injury. It's going to be worse than doing nothing at all. And even a few anti-cancer drugs can induce free radical DNA damage, and that's actually what they're designed to do, so that's a positive way. Now, because we've been talking so much about free radicals, how do we get rid of them once you have them? Well, free radical degradation is produced through enzymes that we already produce: catalase, superoxide dismutase, and glutathione peroxidase. Also by just spontaneous decay and antioxidants. Now, all of these protect you against free radical damage, but remember the catalase, the superoxide dismutase, and glutathione peroxidase. Important stuff there. So now let's move on to infarction. So what's infarction? Well, infarction is tissue death caused by a local lack of oxygen, and this occurs when you get an obstruction of the blood supply. Now, there is something called red infarctions and pale or white infarctions. So red infarcts occur in loose tissues with a lot of collaterals, things like the liver, the lung, or the intestine, or following reperfusion. Now, after obstruction of a blood supply occurs, that ischemia ensues. Now, there's not enough blood supply for the cells to survive, but there are collaterals that ischemia is going to rapidly follow that reperfusion. And because there is cellular damage, that increased perfusion becomes very, very messy. Blood is kind of leaking into damaged tissue, and it's going to cause that red infarct. Now, the other thing that might happen is if you have an area of ischemia, you don't have collaterals, but that ischemia is there only for a specific period of time, maybe just a few hours. And then the ischemia resolves, maybe it's an emboli that's been busted up by TPA or just got rid of spontaneously, and then you have reperfusion of that area that was previously ischemic. Well, there is definitely going to be cellular damage in that area, and that reperfusion also is going to be very messy and cause a red or hemorrhagic infarct as well. Now remember, we just talked about how reperfusion injuries is due to damage by free radicals. So when you reperfuse an area that is hypoxic, you'll generate a lot of oxygen-free radicals, and those free radicals are going to induce even more damage than the hypoxia had induced in the first place. Now let's talk about pale or white infarcts. That's going to occur in what we call solid tissues with maybe a single blood supply, not a lot of collaterals. Think about the heart, think about the kidney, the spleen, and again, sometimes in the liver as well. Again, if you are not revascularizing these areas, you will just end up with a pale infarct. All right, guys, that brings us to the end of the video. It's time for that end of session quiz. I want you to stop the video, try to answer those questions on your own, and then we'll go over them together. First question, what histologic features are seen in apoptotic liver cells? Well, you're going to see cellular shrinkage. You're going to see pycnosis, which is where the nucleus gets kind of condensed and the chromatin is all kind of clumped together. You're going to see karyoresis, which is nuclear fragmentation. You're going to see membrane blebbing, and then the formation of apoptotic bodies. Next, what substances do cytotoxic T cells and NK cells use to induce apoptosis in the cells infected with, say, a virus? Well, they're going to use perforin and granzyme B. The perforin makes the holes, the granzyme B goes in and activates caspases. Next, what highly damaging events can cause irreversible cell injury? So think of things like big calcium influxes. Think of damage to the plasma membrane. Think of rupture of the lysosome, mitochondrial permeability, pycnosis, karyolysis, karyoresis of the nucleus, so any type of damage to the nucleus itself. Next, what cellular enzymes are responsible for handling oxygen-free radicals? So we went over this a couple times. Remember, it's peroxisomes that contain things like catalase, which is an enzyme that degrades hydrogen peroxide to O2 and water. Remember that superoxide dismutase. That's going to be a similar thing. It's going to convert oxygen-free radicals to hydrogen peroxide. And then glutathione peroxidase catalyzes free radical breakdown as well. All right, guys, that brings us to the end of our video. I hope you learned something. I'll see you next time. Hello everyone and welcome to our Step 1 video on inflammation. Now before we get started, I have someone here that I need you to meet. I would be nothing if I didn't have my trusty sidekick and cameraman Travis. Say hi, Travis.
Hello.
Travis is the best of the best. His skills behind the camera are a critical part of the creation of these videos. Isn't that right, Travis?
I guess. Can we just shoot this?
Right you are, Travis. So, how do I look?
Let's see, a little bit to your right.
Okay.
A little bit more. Right there.
Really? I seem to be, I don't know, a little bit not in frame.
I don't know, man. This is looking pretty good from here.
I hear what you're saying. I don't disagree, but maybe, I don't know, here.
Whatever. It's all the same to me.
All right, on to inflammation. Now, sometimes injured tissues undergo necrosis, which you covered in the previous video. But sometimes the body initiates an inflammatory response to try to repair the damage. So we're going to talk about some inflammation. So inflammation, again, characterized by redness, swelling, warmth or heat, pain, and some loss of function. Now, there are two big categories of inflammation: acute inflammation and chronic inflammation. So let's start with acute inflammation first. Now, acute inflammation goes through several different stages. First, you have some type of injury or insult, and that leads to the release of specific cytokines and inflammatory mediators. Now, the inflammatory response is associated with acute phase cytokines. And what are those? IL-1, IL-6, and TNF-alpha. Now make sure you know those. Associated those in your mind with inflammation: interleukin-1, interleukin-6, and TNF-alpha. Now these cytokines do a lot of things, like cause vasodilation, and they increase vascular permeability. So that plasma starts to exude into the interstitium, and that causes swelling and redness and so on. Now, exudation of those inflammatory fluid is going to be measured or mediated by 3 specific chemicals. I want you to remember histamine, serotonin, and bradykinin. So make sure you know those also: histamine, serotonin, and bradykinin. Then eventually you're going to develop fibrosis, where fibroblasts get into the tissue and start laying down extracellular material, specifically collagen. And we'll go over collagen in detail in a later video. But for now, I want you to know that vitamin C is important in this process of fibrosis and making collagen. Then over time, that extracellular matrix needs to be degraded and remodeled into something more organized. Now, the enzymes that do this are the metalloproteinases. They're called metalloproteinases because they require a metal in the enzyme, specifically zinc. So that's another substance that's vital in the healing process, zinc. So you need vitamin C and you need zinc. Now, there are different outcomes of the acute inflammatory processes. Ideally, the normal structure and function are restored, and it's like the injury never happened, and that's called resolution. Now, conversely, you can develop scarring. What's that? Well, scarring occurs when, instead of healing normally, you lay down all this collagen that contracts and forms that scar, and that's going to leave some residual abnormal function. So, for example, if you have an MI, there's a scar that forms in the myocardium, and that scar can't contract normally or relax normally or conduct the electrical impulses normally. So scarring of the heart can lead to a variety of different types of tissue dysfunction and organ dysfunction. Now, sometimes you'll get organization, where you get an organized fibrotic shell that kind of walls off inflammation from the rest of the body. And if that maybe fills up with pus, we call that an abscess. So your body actually considers an abscess a success because you've segregated that infection so that the pus pocket can't cause problems for the rest of the body. And then finally, sometimes with acute inflammation, and maybe you can't get rid of whatever's causing it, then you have that progression to chronic inflammation. So let's switch gears and talk about chronic inflammation. So chronic inflammation causes cycles of tissue destruction and repair, and more destruction and then more repair. Acute inflammation and chronic inflammation are really mediated by different cell types. Now, acute inflammation is mediated primarily by neutrophils, and then chronic inflammation is mediated more by mononuclear cells like macrophages and lymphocytes. And those macrophages are the reason why with chronic inflammation you can have formation of granulomas. Now, granulomas are clusters of macrophages that are found in chronically inflamed tissue. So you can see this kind of central area of macrophages, and then surrounding these macrophages are inflammatory cells like lymphocytes and fibroblasts. Now, sometimes some of the macrophages will fuse their plasma membranes and cytoplasm, and then you get the multinucleate giant cells. So with granulomas, you can see these multinucleate giant cells. Sometimes they're just called giant cells. Now, macrophages start out as monocytes in the blood, but when they leave the blood vessel and migrate into the inflamed tissue, we call them macrophages. And macrophages surround and consume and destroy invading microbes, and they also clean up the cellular debris from all those tissue injuries and inflammation. But when the macrophages don't get shut down appropriately, they keep eating away at healthy tissue, so now they are causing local destruction, and the body has to respond to the damage that the macrophages are causing. So with chronic inflammation, you see this ongoing cycle of tissue damage and then repair. So now let's move on to neutrophil extravasation. What a nice word. So how do these different types of white blood cells get out of the bloodstream and then into the tissue? So we call this leukocyte extravasation. Now, this could be neutrophils, also called the polymorphonuclear cells, or the PMNs, or other granulocytes, or it could be monocytes, or it could even be certain types of T cells. They all use the same basic mechanism. So there are 4 steps that we need to go through. So the first step is that the neutrophil gets sort of sticky and it starts to roll along the vascular endothelium, the inner surface of the blood vessel. Now imagine that you're jumping out of a train at high speed and you sort of kind of bounce and roll along the ground as you come to a stop. And actually, this rolling process allows the endothelium to select for certain types of neutrophils that are going to pass through the endothelium. So this is the selection process, and it's called, funny enough, rolling. Now, on the surface of the endothelium, there are proteins called selectins that are involved here. So there's E-selectin, and E is for endothelial cells, and there's P-selectin, and the P is for platelets. And there's also L-selectin, which is found on leukocytes. So think about this. You can't have these selectins expressed on the surface of the endothelial cells all of the time, or else you would be constantly extravasating neutrophils all the time. Now, you want to have some control over this process. Well, the signal that tells the endothelium to start throwing up all these selectins to flag down some of these leukocytes are those acute phase cytokines that we talked about before, IL-1, IL-6, and TNF-alpha. So these are going to make the endothelial cells, quote unquote, kind of stickier in that area. So they are selecting more neutrophils. Now the next step is tight binding, which involves surface receptors called ICAM1 on the endothelial cells, and then the LFA1 on the leukocyte. Now the LFA1 is part of a family of cell surface receptors called integrins, which helps the cell to attach to external things, like other cells or extracellular matrix. And you can remember that integrin helps the leukocytes integrate. Step 3 is diapedesis. So this is where the leukocyte works its way between endothelial cells and escapes the blood vessel. Now PCAM1 is the molecule involved here. PCAM1 is found on the intercellular junctions between endothelial cells and also on the neutrophils and monocytes and even platelets. And then step 4 is migration, where the leukocyte travels through the interstitium to kind of wherever it needs to go, so the site of injury or inflammation. And we need to remember that these are just cells, right? They don't have a consciousness or a complicated mental capacity. They don't know where to go. They're just sensing all these chemical signals that guide them to where they need to go. So these signals are called chemotactic signals, and they can be like things like bacterial products. So a bacteria will produce substances that have the beneficial effect of attracting neutrophils, but really just beneficial to us, not really so much for the bacteria because we're going to kill it. Now, there are also signals that our bodies make called neutrophil chemotactic factors, which we'll see again in immunology. So there's C5a, which is a component of the complement system. There's IL-8, there's LTB4, and there's also one called kallikrein. Now you should make a note of a couple of other points here. The process of binding, diapedesis, and migration is enhanced by platelet activating factor. And it might seem a little weird that platelet activating factor would enhance leukocyte extravasation, but it also makes some sense as well. So if you have a vascular injury and endothelial injury, you're going to want to activate platelets to stop any bleeding, but then you also need to recruit those leukocytes to help get that little inflammation going and patch up the injury, right? Because again, that's the point of inflammation, to heal an injury or an insult. Now the last point I'm going to throw in here is that there's an immunodeficiency disorder called leukocyte adhesion deficiency. Now from the name you can probably tell that this is a problem relating to leukocyte adhesion. Now specifically, you have abnormal LFA1, so that integrin molecule, and this affects the tight binding step. Now we'll cover this again in immunology, but the classic presentation of leukocyte adhesion deficiency is delayed separation of the umbilicus in newborns, and then again later you get recurrent bacterial infections. And that's because without normal integrins, you can't get leukocytes and macrophages out of the blood vessels and into the tissues to fight infections. Now, there are dozens of chemical mediators that influence inflammation, things like prostaglandins, leukotrienes, nitric oxide, thrombin, and so on. The point is that this is a really complex, tightly regulated process. Now, we're painting an extremely simplified picture of it all. We're just focusing on what you're most likely going to see on your test. Let's move on to transudate versus exudate. So in inflammation, it's going to make the blood vessels dilated and very leaky. And besides leukocytes leaving the circulation and getting out into the interstitium, you're also going to have fluid that does the same thing. Extravascular fluid can be categorized as either a transudate or an exudate, and it's really just based on its composition. Now, transudate is going to be a very watery fluid, and exudate is going to be thick and kind of full of stuff. So think about transudate as being mostly water. It doesn't have a lot of cells in it. It doesn't have a lot of protein in it, and has a low specific gravity. And because it doesn't have a lot of stuff in it, think of it as being maybe transparent. You can see through it because there's not a lot in the way. Now, transudates can form because of high hydrostatic pressure. So pressure is basically pushing serum out of the vessel and into the tissue. Or it can also be due to decreased oncotic pressure. Now, proteins in the serum exert an osmotic pressure that tends to draw fluid in, but if your serum is really low in proteins, then you lose that effect and fluid will leak out of the vessel. So if transudate is watery and doesn't have much stuff in it, then exudate is the opposite. Exudate has a lot of stuff in it, so it's rich in proteins, it's rich in cells, specifically inflammatory cells. Now, one student sent in a mnemonic that the EX in exudate can remind you that exudate has extra stuff in it. So maybe that can help you remember it. Now, exudate can form due to lymphatic obstruction, also inflammation that we talked about so far. And we'll talk about transudates and exudates again when we get to ascites and cirrhosis in the GI section. Okay, so that's it for acute inflammation. Now for chronic inflammation. Remember we said that chronic inflammation usually involves macrophages, and that chronic inflammation is characterized by those ongoing cycles of tissue damage and repair. Now there is a specific pattern of chronic inflammation that I want to highlight called granulomatous inflammation. And this is where you have the formation of granulomas. So what was a granuloma? Remember we said that it's a little collection of macrophages. T cells produce interferon gamma, which help the macrophages to form multinucleate giant cells, which can lead to that granuloma formation. Now, macrophages make TNF alpha, which perpetuates the inflammation and maintains the granuloma. So remember, the interferon gamma acts to form the granuloma, TNF alpha acts to maintain the granuloma. Now, some of the drugs we use to treat chronic inflammatory diseases like rheumatoid arthritis and Crohn's disease, some of those drugs, for example, infliximab, block that TNF alpha, and that's going to cause granulomas to break down. So let's talk about a scenario. If you have latent tuberculosis, where your body sequesters the TB mycobacteria into the granuloma in the lung, then you give that patient, say, infliximab. You tear down the granuloma, what's going to happen? Well, that TB mycobacteria are no longer in lockdown, and now they're reactivated into active TB. So before you start an anti-TNF alpha drug, you always do a TB test to make sure that the patient doesn't have latent tuberculosis. Now let's go through some of the diseases that cause granulomas. Make sure that you know this list because you might see a question where they describe granulomas on a biopsy, or they might show you a picture of a granuloma, and then you'll need to mentally run through the list to see which is most likely given the clinical scenario. So you might want to write some of this stuff down. Now we mentioned that tuberculosis causes granulomas, and frequently with TB, these granulomas are what we call caseating, or caseous is going to mean cheese-like, and that's referring to the kind of gross appearance. You see this kind of yellow white stuff that looks like cheese. It's pretty gross. And then microscopically, in the middle of these granulomas, there's caseous necrosis. So you see this central area of pink amorphous material. Now it's not completely specific, but if you see caseating granulomas, then you should probably think about TB. Now, there are other pulmonary diseases associated with granulomas as well. For example, fungal infections like blastomycosis and histoplasmosis. Sarcoidosis causes granulomas, and we're going to cover that in our pulmonary section. Granulomatosis with polyangiitis, that's what we used to call Wegener granulomatosis. So this is, again, covered in another section, our cardiovascular section. It's the form of vasculitis that's going to cause granulomatosis inflammation in the lung and the kidney. Berylliosis causes granulomas. Who gets berylliosis? People who work in beryllium mines. Churg-Strauss syndrome, that's also referred to as eosinophilic granulomatosis with polyangiitis. That's another autoimmune disease that can result in granulomas. And then moving away from pulmonary diseases, cat scratch disease can cause granulomas, and this is caused by Bartonella hynsula. There's listeria. Leprosy can cause granulomas, and also syphilis. And then Crohn's disease, another form of an inflammatory bowel disease where you're going to get granulomas in the GI tract. So here's a section of colon in a patient with Crohn's disease, and you can see the intestinal glands or the crypts here. But what you really need to notice is this granuloma right here. This is a non-caseating granuloma, so you don't have that amorphous pink center. You do have this collection of macrophages surrounded by inflammatory cells, and then even some multinucleate giant cells. So again, if they show you an image of a granuloma and they tell you that's in the GI tract, then they might be trying to get you to make the diagnosis of Crohn's disease. But if they tell you that the granulomas are in the kidney and the lungs, maybe they're steering you towards Wegener. Or if they tell you that it's in the lung, it could be a bunch of stuff, but maybe they tell you the patient's a migrant worker who's been coughing up blood. That's certainly going to suggest TB to you. So they'll have to give you some clues. Now, there's also an immunodeficiency disorder called chronic granulomatous disease, which of course causes granulomas, and we'll talk about that in our immunology section. And finally, you can see granulomas associated with foreign bodies. So one of the ways our bodies deals with a foreign body is to kind of surround it, engulf it, wall it off with a giant granuloma. So foreign bodies can cause granulomas as well. Next, I want to talk about two important markers of inflammation, the ESR and the CRP. So ESR is the erythrocyte sedimentation rate. Sometimes you'll hear it referred to as the SED rate. So this is a test that measures how fast erythrocytes are going to sediment. So how fast the red blood cells settle down to the bottom of a test tube or a pipette tube. So when you put blood in a pipette tube, inflammatory debris like fibrinogen will coat the red blood cells so that they kind of clump together more quickly. And this will cause them to sediment faster. So in inflammatory states, there's more fibrinogen around. The red blood cells are going to settle faster, and that SED rate is going to be said to be higher. But it's important for you to know that the SED rate is extremely nonspecific. An elevated ESR can mean almost anything. Specific diseases where you almost always see an elevated SED rate would include things like inflammatory joint or muscle diseases like polymyalgia rheumatica, rheumatoid arthritis, SLE, vasculitis diseases, things like temporal arteritis. Infection of almost any kind will increase the SED rate, as well as malignancy and even pregnancy. Now, another nonspecific lab test that we use to detect inflammation is the C-reactive protein, or CRP. So this is another acute phase reactant like fibrinogen, and like the ESR, this is also elevated whenever there's acute inflammation. Now, CRP is made in the liver by hepatocytes, and we think that it's probably going to function to help clear bacteria by binding to microbial surfaces and then acting as an opsonin. So whenever there's active inflammation, you might see an elevated SED rate, you might see an elevated CRP, or there could be other systemic manifestations of inflammation such as fever, leukocytosis, an elevated heart rate, blood pressure, all sorts of things that might mean mediated by cytokines. Now, all right, so that's going to be it for the lecture part. Now it's time for that end of session quiz. Try to answer all those questions on your own, and then we'll go over the answers together. First question, describe the process of leukocyte extravasation. So there are 4 big steps, remember. First, there was rolling, which is mediated by selectins, so they select the leukocytes. And then there's tight binding, and that's mediated by what? The integrins, they help to integrate. There's diapedesis, which is mediated by PCAM1. And then there's migration, which is mediated by the chemotactic factors, things like IL-8, LTB4, C5a, and kallikrein. Next, what cytokines are particularly important in the formation and maintenance of granulomas? So remember, T cells produce interferon gamma. That's going to help those macrophages to form those multinucleated giant cells, which can lead to that granuloma formation. And then macrophages produce the TNF alpha, which helps to maintain the granuloma. Next, what cell type plays a role in inflammation by generating fibrinogen and C-reactive protein? Remember, that's going to be hepatocytes. And the last question here, what cell is most responsible for the acute phase of inflammation? So remember, it's going to be that good old-fashioned neutrophil. So remember, acute inflammation mediated mainly by the neutrophils. Chronic inflammation is going to be mediated by monocytes and macrophages. All right, guys, that's it for the video. I hope you learned something. I'll see you next time.
We've been discussing cell biology, but not everything that takes place in your body takes place inside your cells. So let's talk a little bit more about the extracellular environment, starting with collagen. Collagen is the most abundant protein in our bodies, and it's what provides structure and order and substance to our extracellular space. There are 4 main types of collagen that you need to know about. There are actually almost 30 different types of collagen, but there are just 4 that you're most likely to be tested on. For type 1 collagen, I want you to remember the word strong. So we find type 1 collagen in strong structural tissues like bones and skin, and also the dentin of your teeth. So type 1, strong. You might remember that the word bone contains the word 1. Also wound repair, like scar tissue. Scar tissue is tough and strong, so you really have a lot of type 1 collagen to strengthen that healed wound. For type 2, I want you to remember the word slippery. Type 2 collagen is found in slippery tissues like cartilage and the vitreous body of the eye, and also in the nucleus pulposus in your intervertebral discs. Type 3 collagen is stretchy collagen, so it's found in stretchy tissues like the blood vessels and the skin, and also the uterus, which needs to be able to stretch a lot. Type 3 collagen is also found prominently in embryonic and fetal tissue and in granulation tissue, which is part of the wound healing process. We're going to look at that later. But granulation tissue is beefy red and shiny and often kind of wet looking. Type 3 collagen also makes up a type of filament called reticular fibers, which forms the connective tissue stroma in these organs and tissues. And you can remember it as 3-ticular fibers for collagen type 3. And then type 4 collagen is found in the basement membrane. So again, type 1 is strong, type 2 is slippery, type 3 is stretchy, and type 4 is the basement membrane. So there's an awful, disgusting mnemonic for this, which is to think about a strong, slippery, stretchy BM. You know, BM for bowel movement, but here BM stands for basement membrane. So a strong, slippery, stretchy BM. Now, a less scatological mnemonic is simply the word scab. The S is for skin, which is type 1. The C is for cartilage, which is type 2. The A is for arteries, which is type 3. And then the B, again, is basement membrane, which is type 4. It's not quite as comprehensive a mnemonic as the other one, but it's still pretty decent. So now that we know where we find these types of collagen, how do we make collagen? Well, the synthesis and especially the modification of collagen is about a 3-star topic. About half of you are going to have a question about the post-translational modification of collagen. So here there are 6 main steps. First, the basic peptide is synthesized in the rough ER of fibroblasts. So what are fibroblasts? They're the cells that create scars and fibrous tissue and collagen. So they translate the mRNA and synthesize a polypeptide called pre-pro collagen. Lots of proteins are synthesized as propeptides, and then they're cleaved or modified to become the final protein. But this is actually first synthesized as a pre-pro collagen. This is going to become the alpha chain of the collagen. These alpha chains have a very unique structure. You either have glycine, proline, X, where X is any other amino acid, or you have glycine X hydroxyproline. Now, there can be more than 1,000 amino acids in a row, so these are really long peptides. So that's the first step, synthesis of pre-pro collagen alpha chains. The next step is hydroxylation of lysine and proline residues. This is the step that requires vitamin C. So if you don't have vitamin C, you can't hydroxylate collagen, and basically you stop right there. If you can't make collagen, you end up with scurvy. This is why vitamin C can prevent scurvy, because it's so critical in making collagen. It's also the reason that vitamin C is important for wound healing. Now, you probably remember how sailors would get scurvy because they were stuck on a boat for months and months and months without any fresh fruits and vegetables to eat, and they would become vitamin C deficient. Then once they figured out to bring along some lime juice to make sure everybody on board got a little ration of lime juice every day, they could prevent the scurvy.
It's scurvy.
The third step of collagen synthesis, still inside the fibroblast cells, is glycosylation of the hydroxylated lysine residues. And then you add some hydrogen bonds and some disulfide bonds, and at this point you've gone from that pre-pro collagen to pro collagen. And here we start to see the structure of collagen taking place, which is a triple helix of 3 alpha chains. The fourth step is exocytosis. So at this point, you've done as much as you can inside the cell. But collagen is a huge molecule, so you can't assemble it completely inside the cell. In order to finish the processing of collagen, you have to export the procollagen and finish constructing your collagen outside the fibroblast. The next step then is to process the procollagen into something called tropocollagen. So basically you cleave the terminal regions of procollagen and then you have tropocollagen. That's proteolysis, cleavage of proteins. And then finally you cross-link a bunch of these tropocollagen molecules together to make these enormous collagen fibrils. So this is a covalent bond, a covalent cross-link between lysine on one alpha chain and hydroxylysine on the other. So you do all these cross-links, then you've got your collagen. One thing I want to mention just in passing while we're talking about collagen and wound healing and scar formation, what do we call a scar where there's too much collagen deposition? This is a keloid. A keloid is a scar with too much collagen. And one way we can treat keloids is by injecting the keloid with glucocorticoids, because steroids inhibit collagen synthesis, and that's something worth knowing. Next, let's talk about 3 diseases of defective collagen synthesis. First, we'll talk about osteogenesis imperfecta, then we'll touch on Ehlers-Danlos syndrome and Alport syndrome. Now, osteogenesis imperfecta, or OI, is a bone disease where you have fragile bones. You can guess from the name that you have imperfect genesis of bones. Osteo is bone, so this is osteogenesis imperfecta. Now there are a whole bunch of different subtypes of OI, but the most common type is OI type 1, which is an autosomal dominant disease where you have normal type 1 collagen, but just not enough of it. Or in some cases you might have slightly abnormal type 1 collagen. Either one of those can occur. But remember, type 1 collagen is your strong collagen. It's found in your bones. So if you don't have it, you're going to have multiple fractures with even minor trauma. And and then leg and arm deformities as a result of those fractures. I mean, kids are always falling down and running into things, so these kids are always breaking their bones. And they might even develop bowing of the long bones in utero, which you can see here. They can also have blue sclerae because the sclera is deficient in collagen, so it becomes thinner and it's somewhat translucent, so you're able to see the choroid right through the sclera, which kind of gives it a bluish or grayish color. That's usually a dead giveaway. If they give you a picture of a patient with blue sclerae, they're giving you the diagnosis of OI type 1. Or they might show you an x-ray of a child with tons of fractures in various stages of healing, and you're either going to think about OI or you might think about child abuse. Now, don't mistake one for the other and ship this kid off to CPS or foster care. It could be a case of OI. So how do you tell the difference? You look for those blue sclerae. You might also look for hearing loss because the ossicles of the middle ear can be affected. And you can also look for dental abnormalities because that type 1 collagen is found in the dentin of the teeth as well. Then on the other end of the OI spectrum is OI type 2, which is so severe that these kids die in utero or maybe within the first couple of days after birth. It's sometimes called perinatal lethal OI. So type 1 is much milder than type 2 by comparison. Next, let's talk about Ehlers-Danlos syndrome, or EDS, which is another type of collagen defect. Again, there are a whole bunch of different subtypes of EDS, and review books have been getting this wrong for ages, so I want to break it down and try and get it straight. There's classical EDS, which affects mainly type 5 collagen and also type 1 collagen. Now, we didn't talk about type 5 collagen because it's not one you're likely to be tested on, but type 5 collagen interacts with type 1 collagen while the collagen fibrils are being formed. So a defect in type 5 collagen can look a lot like a defect in type 1 collagen. Now, patients with classical EDS have hyperextensible skin and joint hypermobility. Hyperextensible skin is where you pinch the skin, and instead of stretching like this, it'll stretch like this. And joint hypermobility means that these patients can bend their joints in all sorts of freaky directions, like bending a finger all the way back to scratch their wrist. 100 years or so ago, some of these individuals with classical Ehlers-Danlos syndrome were circus sideshow performers with names like the Elastic Skin Man. Now, there's another type of EDS called hypermobility EDS, which has joint hypermobility without the hyperextensible skin. Now, the exact genetic abnormality for this type has not really been discovered yet. And there's yet another type of Ehlers-Danlos syndrome called vascular EDS, which is due to a defect in the synthesis of type 3 collagen. Now, which type of collagen was that? Strong, slippery, stretchy, BM. So this was the stretchy type. So type 3 collagen is found in the blood vessels. So again, this is vascular Ehlers-Danlos, and it causes arterial rupture and hemorrhages and easy bruising. You can also see intracranial aneurysms with Ehlers-Danlos. So again, blood vessel problems. Now, one last collagen disease to mention is Alport syndrome. Alport syndrome is a genetic defect of type 4 collagen. Which one was that? That's the BM, the basement membrane. Alport syndrome affects type 4 collagen in the basement membrane of the kidneys, which leads to nephritis and kidney failure. We also have type 4 collagen in the basement membrane of the ears, so these patients can have hearing loss. So kidney disease plus deafness equals Alport syndrome. Then also sometimes you might see eye problems, specifically cataracts or something called lenticonus, where you have thinning of the capsule around the lens because the basement membrane of the lens also has type 4 collagen. So if you remember the mnemonic for reactive arthritis, can't see, can't pee, can't climb a tree, you can change that mnemonic around a little bit for Alport syndrome so it becomes can't see, can't pee, can't hear high C. Eye problems, kidney disease, and deafness. So that's it for collagen. Now, what else is found in the extracellular matrix? Well, there's an extracellular protein called elastin. Now, elastin sounds like elastic because it's stretchy. It's even more stretchy than type 3 collagen. So what tissues do we find lots of elastin in? Well, tissues that need to stretch and expand. So it's found in the blood vessels, especially the arteries. The alveoli of the lungs have to be able to expand and fill up with air. It's found in the larynx. And also the spine needs to be able to bend and stretch. So we have elastin in the ligamenta flava along the spine. Now, elastin has a lot of proline and glycine, whereas we said collagen is rich in proline, glycine, but also hydroxyproline, which is kind of unusual. And elastin is non-hydroxylated, so that's another difference between strong collagen and stretchy elastin. Now, structurally, instead of an alpha chain triple helix, we have proteins called tropoelastins that are held together by a matrix of a glycoprotein called fibrillin. And fibrillin is also made by the fibroblast, just like collagen. And if you have a defect of fibrillin, that causes Marfan syndrome, which is characterized by hyperelastic joints, kind of similar to Ehlers-Danlos, and also problems with the heart valves and the aorta, like aortic dissections. Marfan patients are also often very tall, they have really long arms and legs, and they have long spidery fingers, which we call arachnodactyly. So these are the hands of a patient with Marfan syndrome, and you can see that the thumbs are so long that the entire distal phalanx sticks out when he makes a fist. This is called Steinberg's sign. And in this patient, you can see the long, thin limbs and those long fingers again, and he has a chest wall deformity called pectus carinatum, where the sternum protrudes anteriorly, kind of like the sternum of a bird. This is sometimes called pigeon chest. So again, Marfan syndrome is a defect of fibrillin, which makes up part of elastin. There's an enzyme called elastase that's supposed to degrade elastin. And there's an enzyme called alpha-1 antitrypsin that inhibits elastase. Now, this is clinically relevant as well. What happens if your alpha-1 antitrypsin is mutated so it doesn't work properly, so it cannot inhibit elastase? Well, this is a condition called alpha-1 antitrypsin deficiency. Elastase is uninhibited, and it goes around breaking down elastin left and right, out of control. And so with increased degradation of elastin, you're going to get destruction of elastin in the alveoli, which results in emphysema, a pan-acinar emphysema. So if you see emphysema in a non-smoker, think about alpha-1 antitrypsin deficiency, especially a young non-smoker. The other clinical finding in alpha-1 antitrypsin deficiency is that this defective alpha-1 antitrypsin molecule can accumulate and polymerize in the hepatocytes, and that leads to liver disease like hepatitis, cirrhosis, and even hepatocellular carcinoma. All right, that's it for collagen and elastin. Next, we're going to get into wound healing, but first I want to briefly talk about a topic that's related to wound healing that's not really found in most review books, and that's the different mechanisms of angiogenesis or neovascularization. So if you want to regenerate tissues and heal a wound, you're going to need blood supply, right? So there are a couple of interesting ways that this can occur. One way is that you can grow a new vessel branch off of an existing vessel. This starts with enzymes called metalloproteases, which contain zinc. Metalloproteases are responsible for remodeling and reorganizing the extracellular matrix. These metalloproteases can come along and kind of degrade and soften the basement membrane of the existing blood vessel. And then growth factors like VEGF, which is vascular endothelial growth factor, causes the vascular endothelium to proliferate and migrate toward the area that needs a new vessel. Then you have other signals like fibroblast growth factor, or FGF, that recruit supportive cells to come in and lay down basement membrane for this new vessel. The other way to grow new vessels is that VEGF can also recruit endothelial precursor cells, or endothelial progenitor cells, to come out of the bone marrow where they normally hang out, and these cells migrate to where they're needed, and then they can grow new vessels and connect up to the existing vascular bed. So now that we know about how to make collagen and how to make new blood vessels, we can talk about how to heal a wound. For the first few hours after you're wounded, you're going to have hemorrhage and clotting. It may just be small microscopic bleeding, but those ruptured, wounded vessels are going to leak and bleed, right? Then over the course of the first 24 hours, there's going to be acute inflammation. And we said that the cells of acute inflammation are the neutrophils. And of course, those chemotactic factors are going to come into play here to flag the neutrophils down and make sure they get to the right place. Then on days 1 to 3, you have 3 overlapping things going on. The neutrophils are being replaced by macrophages, which come in and clean things up and clear out all the cellular debris. You have granulation tissue being formed, with fibroblasts coming in and laying down collagen, and vascular endothelial cells coming in to reestablish the blood supply. And what type of collagen did we say was found in granulation tissue? Type 3 collagen. And then once you have a nice bed of viable granulation tissue, epithelial cells can start to migrate in. This is called epithelialization. Mobilization of epithelial cells to repair a skin wound. Now this happens pretty early on, and it's clinically relevant because post-op bandages after surgery need to be kept on for at least 48 hours. You're going to keep the bandage on for the first 48 hours so that epithelialization can take place. You don't want to injure that wound in any way or create any sort of osmotic damage with water or soap in the first 48 hours. Now, after 48 hours or so, you might clean the wound occasionally, but in the first 48 hours, you want to let the process of epithelialization take place. Then, over a period of months, you have more and more collagen production. So, at first it was type 3 collagen in the granulation tissue, and now it's more type 1 collagen, that strong collagen that's involved in scarring. So, now we're ready for the end of session quiz. So, I want you to pause the video, go through those questions, and then we'll go through them together. All right, first question. What findings are associated with Ehlers-Danlos syndrome? So with classical EDS, you get hyperextensible skin and joint hypermobility. With hypermobility EDS, you get joint hypermobility with basically normal skin. And with vascular EDS, you get easy bleeding or bruising, arterial rupture, and intracranial aneurysms. Next, what are the different types of collagen and where can they be found? Well, remember, a strong, slippery, stretchy BM. Type 1 is the strong one, found in skin, tendon, and bones, as well as the organs. Type 2 is the slippery type, found mainly in cartilage, the vitreous body, and the nucleus pulposus. Type 3 is the stretchy type, which is found in reticular fibers, the walls of the arteries, the skin, the uterus, fetal tissue, and granulation tissue. And type 4 is found in the basement membranes, or the basal lamina. Next, which amino acids are found in large concentrations in collagen and in elastin? So in collagen, you're going to find lots of glycine, proline, and hydroxyproline. In elastin, you're going to find lots of glycine and proline. And the last one, what's the role of vitamin C in collagen production? Well, vitamin C is important for the hydroxylation of lysine and proline. So if you lack vitamin C, like in a patient with scurvy, there's going to be a defect of collagen synthesis resulting in poor wound healing, swollen gums, bleeding, and anemia. All right, that's it for the quiz, but we also have a couple of rapid-fire facts. Hyperflexible joints, arachnodactyly, aortic dissection, and lens dislocation, this is Marfan syndrome, which was caused by a defect of fibrillin. And then hereditary nephritis, cataracts, and sensorineural hearing loss is Alport syndrome, which is due to a defect in type 4 collagen. Remember, can't see, can't pee, can't hear high C. And then also be sure you can recognize Ehlers-Danlos syndrome and scurvy as well. We just went over both of those. Very, very high yield. All right, that's it for this video. I'll see you next time. Hello there. Today we're painting a portrait of a sweet young lady with osteogenesis imperfecta. What's the classic facial feature of osteogenesis imperfecta? Right, it's the blue sclera, those beautiful blue sclera. So let's take our knife, and we're going to pick up some of our titanium white and mix in a little Prussian blue. Then we're going to get just a little roll of paint on our knife, and we'll go up here to our canvas and just fire it in there any old way. Any old way you want, just like that. There are lots of different kinds of osteogenesis imperfecta, but most commonly there's a mutation in the gene for the alpha chains of type 1 collagen. Or maybe sometimes there's a problem with hydroxylation, but usually it's a mutation in the alpha chains, an autosomal dominant mutation. Looky there. Isn't that beautiful? I think so. It's just that easy. Well, the little clock on the wall says we're just about out of time. God bless you, my friend.
Hello everyone and welcome to our Step 1 video on cellular adaptations. It's time for another introduction. Students, this is my beard. I know, pretty awesome. And while we're on the subject of beards, let's talk about some beard etiquette. Number 1, never grow a beard after your girlfriend dumps you. You don't look tragic or sensitive, just kind of pathetic. Anyway, no one really cares that you got dumped. Number 2, no hipster beards. If you grow a beard because you thought it would look good with your skinny jeans and a faux retro Pink Floyd t-shirt that you got from Goodwill, then you fit the Webster's definition of a hipster. Trust me, it's in there. No need to look that up. Number 3, Step 1 beards are okay, as long as you don't complain about how tired you are. You're growing that beard as a sign of strength, not because you're too tired to maintain a minimum level of bodily hygiene. Send me a pic of your Step 1 beard on Twitter and I promise to repost it. Ladies, sorry if I've neglected you in this intro. Though I would advise against it, you can always scare up some testosterone and grow your very own lady beard, which again, I promise to retweet. All right, let's beard on. All right, so today we're talking about how cells adapt to injury and to stress. Now, in the video on inflammation, we talked about tissue responses, but now we're looking more at the cellular level. Now, we're going to see that cells can become stronger to withstand further injury, or cells can be replaced by new cells. And then we'll look at what happens when cells are irreversibly damaged, and then we'll also look at cells when they kind of get to that give up stage, where instead of fighting back, they just sort of wimp out and atrophy. All right, so let's move on to hypertrophy and hyperplasia. Now, you've probably heard the saying, what does not kill us makes us stronger. And that's actually Nietzsche who said that. Well, how do cells and tissues become stronger in the face of injury then? Well, they can undergo either hypertrophy or hyperplasia. Now, this is really basic, but hypertrophy means the individual cells get larger, and as a result, the whole organ gets larger. So, for example, if you exercise and work a muscle, that muscle will respond by laying down more myofibrils, which makes each individual muscle fiber stronger, and it also makes the entire muscle bigger. So in general, hypertrophy is going to occur due to that production of more cellular proteins. Now, another example would be hypertrophy of the uterine muscle, so the myometrium, and that's during pregnancy. Now, obviously, part of the reason that the uterus gets so large during pregnancy is because there's a big baby in there, but even before the baby gets very big, during early pregnancy, hormone changes cause the uterus to lay down myofibrils, and the cells become hypertrophy, and this is to strengthen that muscle for when it has to contract and push that baby out. So again, this is hypertrophy, but here the stimulus isn't exercise, it's just hormonally induced. So hypertrophy can be a beneficial adaptation, but it's not always. Now, one example that you'll see again in cardiology is myocardial hypertrophy. So there are a couple of different reasons why the heart muscle would hypertrophy, but the most common one is going to be chronic hypertension, where the afterload is increased and the left ventricle has to work harder to push blood against that increased afterload. And so those myocardial cells hypertrophy. Now, this does help the cardiac muscle get stronger and maintain output, but over time it makes the ventricle stiffer and less compliant. So then the ventricle can't relax and fill as well as it should, and eventually this leads to diastolic heart failure. So hypertrophy can be a maladaptive, self-defeating process in many situations. Now let's go over hyperplasia. So what is hyperplasia? How is it different? Well, instead of getting bigger cells with hyperplasia, you get more cells. And when you increase the number of cells, the overall organ can get larger. So the cells aren't any bigger, there's just more of them. Now again, these changes can be triggered by hormones. For example, again, during puberty, girls start producing more estrogen, and this causes hyperplasia of the glandular tissue of the breast, and as a result, the entire organ gets larger. Now, estrogen also causes proliferation of the endometrium as part of the menstrual cycle. Now, we'll talk about this in more detail in the reproduction section, but also remember that progesterone counteracts this proliferation. So if you don't have that progesterone, then this estrogen-driven proliferation becomes endometrial hyperplasia, and this increases, unfortunately, your risk of endometrial cancer. Now, hyperplasia in and of itself is not cancer because it's a controlled process, but anytime you have cell division and cell proliferation, there can be a chance that cancer can develop in that situation. So, some stressors trigger hypertrophy and hyperplasia, but what happens if cells are stressed and injured to the point that they die? How can they be replaced? Well, sometimes they can't and sometimes they can, and that brings us to stem cells. So stem cells are a pretty hot topic ethically as well. So what is a stem cell? Well, basically a stem cell is a cell that can renew itself again and again, and then some of its daughter cells can differentiate into specific cell types, while then again other daughter cells actually retain this ability to self-renew as well. So a stem cell is able to divide into a precursor cell that goes on to be a new cell, whatever that cell is needed in that situation. So think back to the zygote. So it's a single cell. Now if you were to split that zygote in two, would it develop into half a person? Well, no, those cells are still undifferentiated stem cells, so they can keep dividing and they can eventually differentiate and turn into anything later on. And that's why when that zygote splits, if it does, both cell masses survive and they keep dividing and growing and you get identical twins. So at that early stage, every cell has ability to grow into an entire organism. Now let's fast forward maybe a week, maybe longer. You've got a blastocyst. You have a hollow ball of cells with an inner cell mass. Not very many cells yet, maybe overall maybe about 100. Now those cells in the inner cell mass aren't differentiated yet. They still have the capacity to become any tissue in that fetus. Now these are called the embryonic stem cells, and they haven't yet committed to any particular cell lineage. Now at some point those cells do become committed. They'll still be stem cells that will divide and make a lot of different tissues, but they become committed to making just maybe endoderm-derived tissues or ectoderm-derived tissues and so on. Now eventually we have stem cells that are committed to being neural cells or blood cells or whatever the case may be. Let's also think about your bone marrow. It has to be able to constantly make new cells, even now. Now, leukocytes get used up fighting infections. Red blood cells last for maybe only about 120 days under even the best circumstances. So in your bone marrow, you have multipotent hemipoietic stem cells. Again, these are multipotent, so they have that potential to become multiple different types of cells. But they are hematopoietic cells, so no matter what they become, you know it's going to be a blood cell of some type or another. They can't go on to become liver cells or neural cells because they're partially differentiated, but they do still retain some stem cell ability. So here's your multipotent hematopoietic stem cell, and it's receiving a stimulatory signal from the body that says, we need lymphocytes. So this stem cell can grow and divide, and one of those daughter cells is going to differentiate into a lymphoid precursor cell. And it's going to be committed to becoming a lymphoid cell, whether it's a B lymphocyte or a T lymphocyte or maybe an NK cell. That hasn't actually been decided yet, but it is committed to becoming a lymphoid cell. But this daughter cell is going to somehow retain its multipotent self-renewing property. So instead of becoming a lymphoid cell or whatever, it's going to just now grow and divide again and again and again. And that's what makes that stem cell special. Now, one other interesting thing about stem cells is that we've discovered that you can take an oocyte and remove the nucleus, and then if you transfer in the nucleus from another cell, just a regular cell, you can create an embryo with embryonic stem cells that have the donor cell's DNA. And then you can culture those stem cells and get them to differentiate into pretty much whatever tissue you want. So this is called therapeutic cloning, and it's how they've been able to clone sheep and cats and water buffalo and all sorts of things. Now, this is still a long way off from actually being practical, but it's pretty interesting to think about being able to maybe grow new organs for transplant or to grow new pancreatic beta cells for patients with type 1 diabetes or a million other things. All right, let's switch gears. Let's talk about metaplasia. So another way that cells can adapt in response to injury is called metaplasia. Now, metaplasia is a reversible change where one cell type is replaced by another cell type. And that's as a response to some kind of ongoing stressor. Now, there are lots of common examples of this. For instance, the trachea and the bronchi are normally covered with ciliated columnar cells. But in smokers, that normal columnar epithelium is replaced with squamous epithelium because the squamous epithelium is tougher, better able to kind of handle the exposure to smoke and to toxins. But there are potential downsides to this. First of all, there's a reason that we have that ciliated epithelium in the first place. Those cells produce mucus, they trap toxins and microorganisms, and then the cilia help to push the mucus away from the lungs to keep those toxins and pathogens from reaching the lungs in the first place. So when you replace that columnar epithelium with squamous epithelium, then you have less beneficial mucus production and you can't clear it as well either, so you're more likely to get respiratory infections. Now, the second downside is that these metaplastic cells are unfortunately a setup for a transformation into cancer. Now, another form of metaplasia that you should be familiar with is called Barrett esophagus or Barrett metaplasia. So this is where in the normal squamous epithelium of the lower esophagus is replaced by columnar epithelium, which is similar to the epithelium in the stomach and in the intestines. So this is sometimes called intestinal metaplasia as well. Remember, metaplasia is in response to some stress or injury. So what's the injury in this case? Well, in this case, it's chronic GERD, gastroesophageal reflux, where the lower esophagus is exposed to a lot of stomach acid and other gastric contents. Now, the thinking is that the columnar epithelium is more resistant to those acids, but again, with metaplasia, there's a risk of developing cancer. So Barrett esophagus is a precursor to esophageal cancer, specifically esophageal adenocarcinoma. So how does metaplasia happen? Are you somehow changing differentiated squamous cells into a totally different cell type? Well, no, that would be like making a hepatocyte turn into a neuron, so that doesn't happen. Now, what's going on with metaplasia is that local stem cells in the area are being induced to make a new type of cell, although the exact mechanism isn't quite understood yet. All right, so let's move on to atrophy. So we're moving on to another pathological phenomenon, atrophy. So this is shrinkage of an organ or tissue. Now, this is due to reduction in the size of individual cells and or because of the reduction in the number of cells. So it's the opposite of both hypertrophy and hyperplasia. Now, you're probably already familiar with disuse atrophy, so muscle atrophy that results from not using that muscle. So if you've ever had a broken bone, you had to wear a cast for, say, 6 weeks, then you know what I'm talking about. What did your arm or leg look like afterwards when you had that cast removed? All weak and nasty and shrunken. So that's atrophy. So disuse is a cause of atrophy, but there are lots of other things that can cause atrophy as well. Decreased hormone signals, for example, in women after menopause, the vaginal epithelium will atrophy because of a drop in estrogen levels. So the vaginal epithelium becomes thinner, drier, it can actually become more irritated and even bleed and get painful. Again, that's called atrophic vaginitis. Now, you can have atrophy due to a loss of innervation. So if you have a spinal cord injury or some peripheral nerve injury, then the muscle will atrophy and get smaller. Now, an inadequate supply of oxygen or nutrients can also cause atrophy. If you don't have enough nutrients because of malnutrition or malabsorption, then you won't be able to maintain the size of the tissue. It'll get smaller, and that's atrophy as well. And then finally, increased pressure can cause atrophy, and that's due to tissue compression. So for example, if you have an enlarging tumor, it can compress the surrounding structures. It might disrupt blood flow to those structures and then produce atrophy again. All right, so what's going on at the cellular level? Well, initially there's a decrease in cell size and a decrease in the number of organelles. And you're also going to see a reduced metabolic activity. Now, basically, the cell is trying to conserve energy and resources, and there's a decrease in protein synthesis and also an increase in protein degradation. So you're not making more proteins, and the proteins that already exist are kind of getting degraded down, usually by the ubiquitin proteasome pathway. Now, when there's disuse and nutrient deficiency, the cell starts to attach ubiquitin to its own proteins, and then these get dismantled by the proteasomes. And then just like a starving person will begin to digest his or her own kind of muscle for energy and then get muscle wasting, a starving, nutrient-deprived cell is going to really start eating its own organelles as well. It's going to take its own components in a desperate attempt to survive. Now, this process is called autophagy, which really just means self-eating. And most of the digestion is done by the lysosomes, but sometimes the lysosome isn't able to fully digest what it needs to, and then you get residual bodies as well. Lipofuscin is a yellow-brown pigment that forms when there's kind of partial degradation of lipids, specifically by free radical oxidation. So the lipids don't get completely digested and cleared, and then you kind of see this kind of atrophied cells, and they kind of fill up with all these lipofuscin granules. Now, the last major change that a cell can undergo is senescence, or just aging. Now, we said atrophy occurs when a cell doesn't have something it needs, like hormonal signal or a nerve signal or nutrients. But what about cells that do have everything they need? Why don't those cells just kind of live forever and ever and ever? Well, for starters, cells have a fixed number of times that they can divide before they just kind of quit. Even cells in culture that have everything they need, once they reach their limit, they stop dividing. Now, the culprit here is that on the ends of our chromosomes, we have long, non-coding DNA sequences called telomeres. And those telomeres protect the ends of the chromosomes. But every time the cell divides, those telomeres will kind of get clipped off a little bit. So with every round of mitosis, those telomeres get shorter. And basically, the shorter the telomeres, the older the DNA and the older the cell. Now, once the telomeres get too short, the ends of the chromosomes kind of get exposed, and the cell sees that sees that as kind of DNA damage, and the cell cycle gets arrested. So the cell will stop dividing. Now, there's an enzyme called telomerase that can add to the telomeres to keep them from getting shorter. Now, germ cells have a whole bunch of telomerase. Stem cells have a ton of telomerase as well. But in mature somatic tissues, there's virtually no telomerase. So as those cells divide, the telomeres get shorter and shorter and shorter. But where else do we find telomerase? Well, unfortunately, cancer cells. So one of the things that cancer cells do so that they can keep dividing is that they figure out how to start expressing telomerase again so that they can keep their telomeres nice and long. And that makes the cancer basically immortal. And we'll talk about that again when we get to the oncology section. All right, guys, that brings us to the end of the lecture part. It's time for that end of session quiz. Pause that video, try to go through those questions yourself, and then we'll go through the answers together. All right, first question. Does the compensatory growth of muscle fibers occur primarily as a result of hyperplasia or hypertrophy? So if you do a lot of working out, you're going to get more hypertrophy. Next, Does myometrial growth in pregnancy occur primarily as a result of hyperplasia or hypertrophy? Again, the answer is going to be hypertrophy. All right, next question. What can happen to the cells of the lower esophagus in response to chronic acid reflux? Remember, they can undergo metaplasia, and that's moving from a squamous cell to a columnar epithelium, and that's referred to as Barrett metaplasia or Barrett esophagus. Next, what is actually occurring at the cellular level during atrophy? So there's a reduction in the number of organelles, you get decreased protein synthesis, you get increased protein degradation, usually by the ubiquitin proteasome pathway. You can have autophagy, remember that's that self-eating, as the cell begins to kind of digest its own components in the attempt to find more nutrients and then survive. And then there's also lysosomal degradation of cellular components as well. And then finally, what is the lipofuscin granule? So that's a residual body within the cell that contains yellow-brown lipofuscin pigment that comes from incomplete free radical induced lipid oxidation. All right, guys, that brings us to the end of our video. I hope you learned something. I'll see you next time.